history. I've been talking with Josie from Infotech for about the last 6 months about the new treatment that their company is distributing, and it was quite fascinating to me because it was actually full circle. For those of you that don't know, my name is Mike Milano. I'm primarily a flower grower, and I don't know, I guess a hobby vineyard grower, winemaker, I don't know, whatever the heck it is that I am. But I was trained as a bacteriologist at UC Riverside, and my first project there was working with bacteriophages to try and control ornamental bacterial diseases, and I failed miserably. And so when I learned about what Josie had in her team, it was quite fascinating because it kind of came full circle for me. Something that I was trying to do 40 years ago that didn't work, that now has legs. And so as things progressed, we decided that it would be interesting to present their system to as many growers as were interested. And one thing led to another, and then we said, well, Carmen's out there, and Matt's out there, and Philippe's out there, and we decided to pull all these people together to help people get a better understanding of Pierce's disease and the control and various options that are out there available to them today. So this is going to be pretty informal. I think Carmen and Matt and Josie and Israel are open to questions as their presentations go along. We're going to, at the end, try and do a little panel discussion, the 3 or 4 of them, answer any questions that you may have at the end. So I think with that, we're going to get going. If you do have questions, I think we're going to try and get the mics to you. Sometimes it's kind of hard to hear. We'll determine whether that's the case or not as we go along. Okay, very good. So Carmen.
Thank you, Mike. Good morning to everybody. Thank you very much for being with us this morning. And I am really, really pleased that I was asked to participate in this event because I have been involved in the development of this program in the state since the very beginning. So today, my presentation is really a way for you to let you know why it has been so successful. And this has been already about 20 years of constant pulling a lot of strings from so many different parts that have been cooperating in a good way to have the result that we have today. So in this picture on the back with all my information, you kind of blurry, you can see a vineyard. And a concept of that, and that's the way I wanted to start this presentation, was for you to see that that was a change that we had started seeing in the late 90s and early 2000s. And suddenly the growers in the Temecula Valley start seeing a lot of vines dying, and they didn't know what it was. Some of them thought that it was probably a lack of water, because the symptoms of this disease really mimics some dryness. So they start adding a lot of water and nothing happens. And suddenly in this field, you can see that half of it is pretty much with a lot of scorching and the other half pretty much not. We had no idea at that time what was happening. And over the years, we found so many interesting things. And one of them was that certain varieties are more sensitive. So this starts really moving very fast. And you can see that after having the vineyards, in a few years, a lot of the growers were really forced to remove them. And it was really sad to see in the valley those piles of vines because they were not producing and they were dying very fast. It was really concerned if this going to be spreading even more. And that really inserted a lot of energy. And in the news, everybody was talking about it because we didn't know what was happening. It was this concern that probably Temecula Valley wouldn't survive. So that really put a lot of energy that made a lot of people, institutions, and different parties to work together. And the main thing that I think has been essential for this to be successful is that we all have a common goal. That really is the core of this partnership that we started many, many years ago. And this is no news for you, just to go through what the success of this program has had, that has been really the ingredient to have the success that we have. This program has been even duplicated in other countries. I was asked to go with, who developed it in California, Dr. Nick Toscano, in Chile. And we provided the resources with the information that we had, and that was mimicked over there due to the success that we had. So coming back to the partnership that we have had, having this common goal that we needed to know what was going on, and they're really interested to maintain the industry in the area. So this really made able to have teams from different agencies, universities, locals, and state dependencies, institutions that really brought a lot of talents and brought a lot of resources that made us all work together. This really accelerated the scientific progress among the researchers, because that really invited us to have teams, and we were participating with different strengths, different abilities, and different knowledge, and different skills that really made the main force to the development of a lot of research that was really needed. And in turn, this really enhanced the relevance of the research. The researchers were not just trying to understand different factors or different things. It was really the interest in a very, very specific purpose. And one of them was to understand what was causing it, why, and what could be done to prevent it. So we're different areas that were really incentivized to develop a lot of research. So this really made to have a collective effort to have the investment of the resources with knowledge and the participation of the different areas to make this possible. And not only that, not only the researchers or the leaders of the different institutions were benefited, it was also an opportunity for all the different staff and the employees that were participating to have more opportunities to grow. So once we had that, all kinds of ideas came up, and we were participating in trying to understand where it was the problem, what was happening. So there were very basic questions that needed to be asked. And essentially, this was a huge effort with the wine growers in the area. So to do that, we needed to have funding. And it was a group very active with that developed a plan, and it was presented in Washington, and we received federal money. It was also money from the state and local government. The growers assessed themselves to have funds to fund the research, and other industries participated as well. One of them that was really hit was the landscape industry, because we found that the insect that was the vector that was discovered, that was the cause of moving the disease, were really, really affected because it was restricted to move the plants. So this industry also had an interest in participating to understand and prevent the problem. So over the years, since 2001, this is the amount of money that have been invested. I don't think there is any other insect, and I might be wrong, but I don't think there is any other insect that has been understood and researched as much as the glass green sharpshooter that is the vector of this disease. that you can see here all the amounts from different areas that have been supported over the years to really bring this knowledge and support the research to do that. And recently, not only that, it has been expanded to other threats that the industry is suffering, as we have other insects and also other diseases that are really a threat for the industry, not only in the area, but across the state. And one of the successes, I just took this picture from one idea that at the very beginning seemed so distant, that it was the development of resistant varietals for Pierce's disease, that is the disease that is caused by this insect. 20 years ago, it was kind of a dream. How would this be achieved? If that would be possible. So the time has passed, and that recently, those are 5 different varietals, 3 reds and 2 whites that are being with patent developed in process. And this research was developed by Andy Walker at UC Davis, and it has inserted genes, not inserted, selected genes from natural vines that have the quality to make wine. He has been providing the wine in different areas, has been tested. I participated in one of them. And you cannot tell, you are not aware that this one of those resistant varieties. It has been really welcome, and this is going to be huge in the areas that have no other ways really to prevent this disease and these sectors that continue with the trend. So I would like to end just to give you a testament that Karen Rose, the Secretary of the California Department of Food and Agriculture, made it in the 2021 report. And she said, what we have all gained the past 20 plus years is confidence. From detection and response to communication and research. This is a mature, effective, prudent, and exemplary program. This is a testament of the power of a true public-private partnership. And the credit for that goes to the growers, the listeners, the stakeholders who have created and sustained. And voila, here you are again, participating in the opportunity for us to let you know there is another tool that probably will bring us some new ways to face this threat that we still have to the industry. With this, I will end, and I will ask if you have any questions, or we can follow with the program. Thank you.
Any quick questions for Carmen? All right.
Thank you, Mike.
If nothing else, then we have Matt Dockery from UC Riverside, your specialist, right?
Yes.
The official title.
Cooperative Extension Specialist.
So Matt has been doing work for a while, tracking and monitoring for Classic winged sharpshooter, which is the primary vector of Pierce's disease. It's the nasty little bugger that moves this disease around. So with that, I'll let Matt take it and give you the lowdown.
Hey, good morning, everyone. First, thanks to Mike and Taylor and others who made this happen this morning, and also Carmen for kicking us off this morning. I'll actually kind of pick up where she left off in some respects. And a lot of what I'll focus on relates, as Mike just said, relates to Classic winged sharpshooter, the primary vector here in, I'm pointing at myself. There we go. Here in Southern California vineyards. And I'll spend a bunch of time actually going into kind of more of the background of what we understand about the biology and ecology of this insect, to really give you some perspective on where the management tools kind of came from, kind of where these strategies, how they were developed and why they were developed. Okay, the first point to make though is it's not just Classic winged sharpshooter here in California. We have 6 or 8 pretty common species. They vary a lot regionally in terms of which ones are the most important. For us, it's mostly about glasswing, and we'll focus on that today, just in the interest of time. We do have a little handout up here. If you guys want, it has a little bit more information about some of the other vectors. You will see some of these other ones. We won't spend too much time on unless you guys have questions about that. So I'll take a look there. Also, while I'm at it, there's at least a handful of these PD booklets put up by UCANR, have some nice photos and a fair bit of background information on this disease, including a little bit on sharpshooter management. I don't know if I've brought enough books for everyone, so you're going to share. So again, we'll spend most of the time though really focusing on glasswing sharpshooter, because it really over the last 20+ years has been the dominant vector here in Southern California. The thing when you get a new vector, it is moving into a system, invading an area that already has these existing native vectors. It begs the question of kind of what is this new vector doing? And so once it became clear that glasswing started to cause all these problems in Southern California in the late 90s and 2000s, it kicked off a whole bunch of research to understand what is it about glasswing? Why is it special? How is it causing these problems? More so in some cases, at least here in Southern California, more so than the natives do. So I actually spent a fair bit of the early part of my career doing just that, trying to understand the nature of glasswing's impact. So the obvious place to start with this, and that we did and several other researchers, is okay, let's see how efficient this insect is. And what I mean is, like, if you put it on an infected plant, how often does it pick up the bacteria? And if you put it on a healthy plant, how well does it inoculate or infect that plant? And it turns out not very well, fortunately, at least compared to some of the natives. The blue-green sharpshooter, I'm showing you some data here, is 30 to 50% more efficient at transmitting to grapes than is glasswing. We actually did what's called a meta-analysis, where we took the results of a bunch of studies, kind of combined them together and estimated sort of independently the efficiency of these two vectors, and found that glasswing is actually way less efficient than the native blue-green sharpshooter. So that's kind of good news. It's not that this new insect, glasswing, new at the time, I should say, is especially efficient on sort of on a per insect basis. We followed that up with a series of surveys, as well as some studies like this cage study, to estimate kind of how well this insect kind of moved the pathogen. There's reasons to believe, based on its biology, really its behavior and its movement, that it may be just better able to kind of move the disease or move it further distances than the natives. So we tried to get at that. Long story short, that doesn't seem to be the answer as well. It does spread the pathogen, as you might expect. Maybe at certain times of the year, it does a little bit better than the natives, but it's not overwhelming. It's doing it. So it doesn't seem to be the mechanism by which it's causing problems either. And so really what it boils down to is based on a bunch of survey work now, is that you get problems with PD in areas where you get persistently high glasswing populations, so places next to citrus and other reproductive hosts, ornamental plantings, things like that. So it's really glasswing's ability to achieve these way higher densities in the native vectors that allows it to have this impact. And that's kind of counterintuitive, but that's good news, right? Because the abundance of an insect is kind of one thing we actually sort of have control over, because these IPM tools that we can use against it. So it's not the end of the world if it's impacted mostly from being able to be really abundant. So I just gave you 10 years of my career in 3 slides, which is a little bit depressing. But I want you to have an understanding of it. Basically what I'm trying to get at here, give you perspective on, is glasswing's it's not the end of the world having it here. We are able to deal with it as long as we stay on top of it. So the partnerships that Carmen talked about are important, will continue to be important, but it is something that we actually can tackle. And there's evidence of that. So early on in the sort of after some of these interventions were put into place in the late 90s and early 2000s, as Carmen mentioned, it became apparent within just a few years, actually, that glasswing populations were on downward trend. And so this graph here is by one of my colleagues, Mark Hoddle, who did 10 years of visual surveys on I think a bi-weekly basis in the citrus grove next to UCR campus, where he surveyed for adult glasswing as well as egg masses, and then kind of kept track of that over this 10-year period. You can see there's a really sharp decline between 2002 and 2012. It's more than 95% reduction. So something's going on, right? Presumably it's at least part of these control measures that are put in place.
Amen.
Yeah.
just a comment for those people that are on Zoom, if they have questions, to put them in the chat box.
Did you guys hear that? Those of you on Zoom, if you have questions, please type them in the chat box and we'll read them out. Any questions? Okay, you guys feel free to in the room here to jump in as well.
I have a kind of quick one here.
Yeah,
This data is for near UC Riverside campus.
it is.
So how does that translate to the areas around that? Is it?
That's a good question. So the best information we have, well, another source of information, this comes from the trapping data in Temecula. So we see similar trend. We don't have it temporally resolved the same way. But on the bar on the left there is trapping, sticky trap data from Temecula in the late 90s. And then on the right, it's from 2014, which is what we did. We went back and resurveyed basically 15 years later. I don't know if you can see that, but that's like hundreds of times fewer insects. So we don't have, to your question, we don't have statewide monitoring per se, although we do in certain regions in these air wet treatment and monitoring programs. Outside of those, we don't necessarily have information on it, but from what we can tell, all the trajectories were somewhat the same. If you look at this data, and I'll come back to this in terms of San Diego because we don't have a great source of information for you guys. Unfortunately, the closest we have is Temecula. That makes sense. Okay. So if insect populations are down, give me 1 second. Okay. If insect populations are down dramatically, presumably that translates to some benefit in terms of reduced PD, right? And the context here is kind of going back to what Carmen was saying in the heyday, the early days, the late 90s and 2000s in Temecula. It's estimated probably 30, maybe even up to 40% of the vines were lost or pulled out because they were starting to show symptoms. That's a staggering number, right? That's thousands of acres of grapes in just a few years being killed. We went back again, much later, 2015 or 2016, and resurveyed 30-something vineyards in Temecula, see where the current situation looks like, and found that PD was way lower. So across those few dozen sites, it averaged about 0.5% of the vines were showing evidence of PD. And I don't know if you can see those little bars or each of the different sites. The majority of the sites we surveyed had no evidence of any PD vines at all. So a total, turnabout compared to what it was like 15 years ago. Again, not surprisingly associated with sharp declines in glassing. So you had a question?
Yeah, so you were mentioning earlier about the blue-green sharpshooter.
Yes.
That, if I heard you correctly, that actually can transmit the Pierce's disease faster and more effectively than the glassy wing, but the glassy wing can transmit it farther than the blue-green.
Yeah, so the question for those of you on Zoom is blue-green sharpshooter and sort of its ability to transmit relative to glassing. And yes, so all available evidence is that the blue-green sharpshooter is much more efficient. So an individual insect can inoculate far greater rate than glasswing. Glasswing is active more over the year, flies greater distances and stuff like that. So there's concerns that it can actually spread the disease a little bit more. We didn't find that to be that's somewhat true. It wasn't overwhelmingly the case. The bigger point is that glasswing can achieve densities hundreds of times higher, at least it did back in the day, early on, hundreds of times higher than blue-green sharpshooter. And we do, just for the record, we do have blue-green sharpshooter down here. It's distributed throughout the Coast Range. Down here in Southern California, it tends to be really coastal though. Up north in Napa and Sonoma, it gets in a little bit more throughout the Coast Range. Here, it's sort of on the ocean side of the hills around here, but you will see it occasionally. It's just, its populations are far dwarfed by glasswing in a typical year. Cool, okay. All right, so the point of all of this, right, is that effective management of PD via controlling sort of regionally glassing is achievable as long as we kind of stay on and we keep this partnership going and keep going, keep attacking. Yeah, question.
We have a question from online. It's what are the factors on the x-axis of the graph? Are those cultivars, grapes, or locations?
Yeah, so the question from Zoom is it looks like you guys can probably see it, but what's on the x-axis here, and those are different sites. I just showed you sort of a histogram of the 30, I think it's 30 something sites in Temecula where we surveyed PD. And so the main point I was trying to show there is there's one site, it's essentially an abandoned, unmanaged vineyard that had 5% PD. About almost 2/3 of the sites had 0 PD that we could detect. Okay. And I'm happy to make this available if you guys want, let me know, and I can send out a PDF to folks. Okay, so effective PD management is possible if we can keep glasswing population under control, which we think we can do, at least in many cases. So Carmen touched on this sort of steps that were taken early on to deal with glasswing. And a lot of this boiled down to these area-wide control programs in Temecula and Kern County, Tulare, and then on up to Fresno. And the goal in those area-wide control programs is to do these large-scale monitoring and as well as management, primarily in citrus. And the reason being is that citrus is this really key host for sharpshooters, especially overwintering hosts. So the idea is to knock down the populations there with biocontrol and chemical control in citrus before it moves out into grapes. So that's a big part. And this is, just keep in mind, this is all kind of going on around you guys, around your vineyard. It's not something you've necessarily seen, but it's been an important part of the response to glasswing for 20+ years now. The other part, a little bit closer to home for you guys, is at the same time management steps being taken in vineyards or immediately surrounding them. So if you look at this PD book I sent you, or the UCIPM site for sharpshooters, you'll see there's a range of steps for recommendations for managing sharpshooters as well as piercing disease. I'm listing just a few of them here. I'm going to spend time really honestly briefly going through just the first few of these related to monitoring and control, mostly just focusing on the insect. The other part kind of relates a little bit more to disease side of things. Maybe save that for discussion for another time, or the later speakers may touch on it. So yeah, anyway, those are some of the things going on in vineyards to deal with glasswing and reduced PD. So before we get to that, the details of that, if you ever see me give a talk at all, I probably showed a graph like this. I have a little bit of an obsession with these seasonal counts of insects over time, because I think it actually gives us important information about the biology of these insects. And so what's being shown here is, this is from the Temecula area-wide program. So bi-weekly or monthly counts of glasswing over the course of the entire year, right? And each line here is a different year going back. I think it's 2007 or maybe 2006 up through 2023. The blue line here, if you can see it, the blue dash line is the historic average. And so I put this up for a couple of reasons. The first of which is hopefully you can see that there's a ton of year-to-year variation, right? Some of those gray lines are hitting up 1,500 at the peak in July. Some of them like 2012 and 2013, maybe 100 at the peak. So like 20-fold, 25-fold difference in insect abundance. We don't fully understand what it is every year, why glasswing populations can vary so much year to year. And as a result, and I should point out that there's also a ton of spatial variation in where you find glasswing being active, and it's not always predictable. And I bring this up because it really, that last point there, it is important. That monitoring, that means that monitoring is especially important. Just keeping track of where the insect is, when it's active, stuff like that. It really directly feeds into management and need for management in your vineyards. So don't neglect that, and I'll come back to monitoring recommendations. The other point about it, though, is the seasonal patterns are actually a little bit more predictable, more so than the year-to-year patterns. So there is some variation, but usually what we get happening is things are quiet this time of year in terms of glasswing. You don't get too much activity. And then starting at the end of spring, sort of end of May and early June, we'll start to see it tick up a little bit. And that red box is kind of when things go nuts. And that's sort of like June through August, in most years, there'll be a huge increase. And that's sort of that early summer primary peak that we see in pretty much every year. And the timing of it varies a little bit, but usually that peak is occurring between July and early August. In most years, that declines pretty sharply at the end of August and through September, October, and it goes back down to really, really low numbers. Occasionally we will get, and it's illustrated in that orange box there, we will get sort of a secondary peak, usually in September. It's about every 3 or 4 years. And again, we don't fully understand why that is. It happens some years and not others. But you will see glasswing kind of rearing its head again. But in all years, we see at least some primary early summer peak during that red period. And I point this out because a lot of what we're doing in terms of management, especially if it comes to insecticide programs, is trying to knock down that early summer peak. So that the timing of the activity of the insect very much dictates the timing of where we're, or when we're implementing, control measures. Okay. Yeah, question.
Could you clarify a little more when you seem to indicate, as far as monitoring, you can't really make any predictions where you see the infestation? It seems to me there had to be some sort of general rules. For example, my vineyard's at 3,700 feet.
Yeah.
I'm 20 feet as a crow fly from the nearest citrus grove.
Yeah.
I would think we'd be less likely to see it, right?
Less likely?
Yeah.
Well, because of higher elevation, but if you're right next to citrus.
No, 20 miles.
Oh, 20 miles, I think you said, okay, sorry. Yeah, so that's what I was saying, there's spatial variability and a lot of it, especially early on with proximity to citrus. To your point, once you get a handle on your vineyard, I think you'll get a sense for kind of where you're gonna see, especially a big block, you might see more insects in one area of it than another, stuff like that. And you see the time of year, things like that. Until you have that information sort of built up over time, it's guesswork unless you're actually doing that monitoring. I guess that was the point I was trying to make. And even so, there will be variability a little bit, especially year to year in terms of the number of when they're showing up a little bit in your vineyard. And I'll come back to this when we talk about insecticide programs, especially organic treatments. Because of the short-lived nature of those treatments, that timing is really, really important. And so if you probably have a decent idea if you've been doing some trapping or other monitoring or when glass swings out there and where, staying on it though is really important. That was the point I was trying to make. Yeah. Other question?
Also hosts to it.
Avocados they're listed as one of the 200 plus hosts of glass swimming. In my experience, they're certainly not as much as citrus. Olives probably up there as one of the better hosts. And there's a pile of ornamental kind of residential or even, the common ornamental around wineries, for example, we see glasswing. Nothing as much as citrus though, at least at that scale of big ag. And the problem is, right, is we grow citrus next to grapes here, unfortunately, too often, so.
Can you have that second peak more than the first one?
No, I've never seen that actually. In fact, and it's not broken down by year here, but the first peak is always bigger than the second one in those years where we see a second peak. So that's why it's a little perplexing, but it's definitely not like a super delayed year where that second peak is actually the first peak's delayed. That's what you're saying. They seem to happen together when they're happening. Okay, any other questions? I don't want to take up too much time out of the program, but anyway, I will try to field these questions as they come. So I mentioned San Diego. So in case you can't tell, a lot of what we know about glass swing in Southern California comes from the Temecula area-wide program, which has been going on for 20 plus years now, since Carmen said. So as I said, a lot of the timing of stuff and other information we know about glass swimming is based on that. We don't really know necessarily a whole lot about glass swimming outside of that area, at least in Southern California. And so fortunately, I was able to sucker at least a couple of you guys in the room, including Mike, and to helping us fill in this gap. And this is something Carmen and I have been worried about for years, right? Is to what extent is the Temecula pattern sort of generalizable to other areas of Southern California? And so we got maybe 24 or so, a little more than 24 of you to contribute sticky trap data, basically the same way we're doing it in Temecula at the same time. So we get at least a little bit of an idea of what's going on in the sort of the northern part of San Diego County down. I think there's a couple of property owners down about where we are here, nearby where we are in Escondido, on up through a little bit north of Fallbrook. And that's shown here, the blue lines, the San Diego data, the red lines, Temecula. This is 2021, which is a kind of a weird year, relatively low year in Temecula. But you can also see there is that secondary peak occurring. So there are some differences there between Temecula and San Diego, for sure. What's reassuring to me though, is that the timing of those peaks and activity kind of match each other. They track a little bit. So hopefully that means, and again, it's only 1 year, but at least we know in that year, Temecula is somewhat representative of what's going on in San Diego. So, but again, not a total substitute for you guys doing your own monitoring. Okay, so now we'll kind of get into the management strategy part of stuff. And the point I want to make first is clarify what our goal is. And I kind of touched on it in terms of knocking down that peak period of activity for glass center. So really, we're trying to suppress that time period. We're not trying to essentially eradicate or keep things at zero over the entire year, especially in the later part of the season, which is a little bit of a benefit to us. And the reason we don't necessarily worry about things after August, certainly into September, October, is this cool, interesting quirk of the PD system where it turns out grapes, when they get infected, actually can lose the infection over the winter. And it seems to be very much tied to when they first became infected. So if they get infected March, April, basically there's plenty of time before winter for those infections to get roaring in the vine. They stay chronically infected and eventually gonna die unless there's some kind of therapeutic that you can knock out or reduce the infection. So early in season infections are bad. Very end of the year, it's not the case. So like start after, it's a little hard because these are numbers, these are day of the year. This is, I should point out some work by folks at UC Berkeley back in the day where they actually inoculated vines at different times of the year. And followed the fate of those infections through the next year. So starting, it tails off pretty sharply, maybe end of June or something like that. But when you get into certainly the end of July and early August, you can see that the vast majority of those infections that are occurring at that time of year are actually recovering over the winter. And so really what we're trying to do is keep glasswing down during that time period when those infections are likely to develop to become chronic. If the vine gets infected because there's a few glasswing out there in October, we don't really care probably because those infections are very likely to recover over the winter time. So they're that dead ends, those are dead end infections at that time of year. And that gives us a little bit more breathing room because we don't have to be crazy aggressive all year round, right? So here's my cartoon of what the management programs look like, at least in terms of the insecticide part of things. This is for like a caricature of a conventional program, where really the backbone of conventional programs here in Southern California are built off of the spring application of one of these long-lived systemic immunics, or it's like metocloprid or something. I'll talk about the details of this in a minute. And the point being is if you apply it, say March, or hopefully not too late into April, you get 3 or actually probably upwards of 4 months in many cases of high levels of insecticide residues in plants enough to kill sharpshooters. So that carries you through that first early summer peak. Occasionally, like as I said, if we get a secondary peak in September, it may be necessary, and it's sort of up to the discretion of growers to do an additional, probably foliar application of a pyrethroid or something to knock down the insects in September, which is then kind of bordering on when we really think we need to knock down the insects anyway. That's sort of what the plan looks like, sort of this long spring application of a long-lived systemic followed by perhaps in some years, an occasional year, a shorter-lived foliar application. The organic programs look kind of similar in terms of the timing, right? But the difference is that the applications tend to be much more short-lived, as I'll explain in a minute. And so as a result, when you start them, and the number of them will really depend on what the monitoring says. So it's really important to stay on top of the monitoring because these things only last very short periods. And so typically you have to do a series, maybe 4 or 5 applications, which is challenging and gets expensive. And obviously need to be done right. So that's where monitoring comes in. But they're both, both programs are trying to do more or less the same thing temporally in terms of the time of year we're worried about things happening. Yeah,
Are you talking about the metocloprid
as a systemic immunicide, yeah.
is it really an insecticide? I thought it just made it distasteful and they would go to another
It
plant.
does both, actually. So the questions of metocloprid, and it's absolutely insecticidal.
It is, okay.
And it is so it takes about a week or 2 for uptake into vines, but then after that, there's some nice data showing 3 months, oftentimes upwards of 4 months of residues. We know what the target is. It's about 10 parts per billion, and vines can maintain that for 3 or 4 months. At least here in Southern California, they take it up pretty rapidly, and most in our soils because they're pretty sandy. There is a sort of secondary benefit of metocloprid where it has this anti-feeding effect. It has like basically an astringent thing where it's distasteful to them and they will shut down their feeding.
The late season application, doesn't that kind of stay around for harvest then? Isn't that a concern?
No, so that's the point, is that it should be wrapped. So a late season application, absolutely. And so I'll come back to that, especially with the pyrethroid. If you're thinking about doing an application of something in like end of August or September, you got to work around harvest in terms of post-harvest intervals. And so there's a big issue with timing there for sure. which is like something that, obviously best avoided if you don't have to, that's why. Again, like if you're not seeing that big peak in September, why bother doing anything about it? You should be okay. Okay, so I'll spend just a couple of minutes on just, because I've been trying to hammer home the importance of sharpshooter, monitoring for sharpshooters. And so the main way we do that is with these sticky traps. Multiple sticky traps distributed throughout the vineyard, typically on the edges is what I recommend. And if you have an idea about sort of the most local source of sharpshooters, like if you're near citrus grove, maybe prioritizing trap placement there, but really distributing throughout the edges as well. Or ornamental plantings, like if you're on the edge of a pretty big residential area, or if there's if you have a winery and significant planting around the winery, it may make sense to prioritize trapping there. These traps are not inherently attractive to sharpshooters. They don't have like a pheromone or anything. So it's kind of a more is better thing, unfortunately, up to, whatever's reasonable. So usually what I recommend to folks is if you have a little block, like an acre or something, start with at least 3 traps and then add more for larger acre blocks, maybe another trap every few acres beyond that. So for like a 10-acre block, you might have 5 or 6 traps. And then making sure they stay up above the canopy, just so there's straight line of flight for the insects to smack into them and get caught. If they're buried down in the vine, you're not going to catch anything, which is kind of a headed and sand approach. And then the final point is checking them as frequently as is reasonable. I usually recommend kind of twice a month during the growing season, bud break to at least harvest. But then even a little bit over the winter and this time of year is helpful. There's not a ton going on right now, but doing at least checking once a month can be helpful. And the reason being is these early times of the year, it turns out, the counts we get this time of year, although they tend to be low, they're kind of predictive of how bad things are going to get later in the year. So keeping an eye a little bit on even on the off season is not a bad idea. And then for the overachievers in the room, sweep net sampling is definitely another way to monitor. It's even more important for some of the other vectors, spittlebugs, and some of the grass feeding sharpshooters that don't tend to pop up like that. So something to consider, especially if you guys are seeing them or think you're seeing them, keeping an eye on them with sweep net sampling. And the real anal retentive folks will actually do visual inspection, especially for young vines. You can actually, when you're say scouting them for PD in the summer, it's pretty easy to see glasswing adults as well as egg masses on them. So, spend a little bit of time. As vines get older though, it becomes, prohibitively time consuming to try to actually visually inspect each vine. The canopy is just too big for it to be practical. Okay. So I'll touch quickly on biocontrol, which is kind of going on around us, which is the cool thing. And this kind of goes back to the airwide programs that we've touched on a little bit. There are, it turns out, multiple species of both native as well as introduced tiny little parasitoid wasps in the family Mimeridae. The main genus is Cosmocomodia. It used to be Gonatoceras. They changed the name. This is a program that's been going on for 20 plus years by the CDFA. Using kind of initial research by folks at UPR, they had identified these wasps, in some cases brought them in from other areas of the US, did a bunch of testing and then handed them off to the CDFA. And so they've been releasing over the years, as you can see, millions of these parasitoids in areas where glasswing is present. And they're currently releasing 3 species. And the most kind of important or dominant one is this Cosmocomodia ashkita. These are great little parasitoids that attack the females lay the eggs and the egg mass the glasswing. You can see that on the top photo. The egg of the parasitoid will hatch. The larvae will eat its way around, kill the developing glasswing egg/nymph, and then they'll leave you the emergence hole. So in that, hopefully you can see that middle photo there. Those little round emergence holes are the parasitoid wasps, the next generation adults coming out. So if you see these little brown scars on their leaves with the little holes in them, little round holes, that means there's biocontrol agents doing the thing. And by all standards, they've had a really big impact, we think, especially in these areas that are kind of mixed ag residential. Yeah.
Is CDFA releasing these down here?
I'll come to that. Unfortunately, they stopped. They stopped recently. So I showed you a different version of this data before. So when Mark Huddle's tracking and glasswing in the citrus grove and citrus, he's also keeping track of parasitism. So over that 10-year, 95+% decline in glasswing, we saw regular parasitism going on with egg masses, 35% on average and upwards of 80% in some cases. So pretty big impact, right? 80% is no joke. There's other data. the CDFA is collected that shows similar things seasonally. So these parasitoids take a little while to get going. This time of year, they're not as active, but then starting late spring and certainly through summer, they start really aggressively attacking glasswing, upwards of 80% parasitism. So by all indications, they have had and are having a big impact on glasswing. And part of the reason we think we see those huge declines compared to the early days. To your point, Mike, they have been doing these releases throughout of these 3 species. They're still continuing in the Central Valley. They stopped unfortunately in 2012 down here. My understanding is it basically comes out of a budgetary thing, but also part of it was essentially our populations of glasswing down here have been pretty low and sustained low. There haven't been any obvious epidemics of glasswing, so they use that as a justification to cut costs. This is something that actually Carmen and I are hoping to chase down a little bit though. I'd like to verify that the parasitoids here in Southern California still are pretty abundant. All indications are that they are established and they are still having an impact, and CDFA up until about 10 years ago was also doing monitoring to confirm that. We just haven't seen anything about 10 years. And so I personally just want to make sure. So I bring that up because I may come looking for cooperators to let me poke around their vineyards.
It must be in the citrus grove.
Yes. So yeah, the question is in citrus. Yeah, absolutely.
What's the status of imidacloprid? Is it gonna be banned in California sometime soon?
There's 2 issues with imidacloprid. One is that, and this CDFA, DPR, California Department of Pesticide Regulation are looking hard. And they're trying hard to evaluate that. It's unclear at this point.
Cause you have some late season exfoliar application, that's not good for bees.
No, and so all these things, that's a good point. All these things you have to work around. Flowering times, pollination. So doing applications even in the spring and like citrus growers, they're having to work around that for sure. The other part about the other issue with the imidacloprid recently, are we good or?
I think we're back.
Okay. The other issue with imidacloprid is there's been some issues with insecticide resistance developing of it. They've seen it up in the Central Valley in pockets. Fortunately, the available data so far in Southern California, I think we're good in good shape. So, and again, I think it probably comes back to the fact that there's less extensive continuous application over huge acreage down here. It's much more patchwork, which shouldn't allow resistance to develop. Okay. Yeah.
Do you think you could control the insect population without using insecticide and just using the biocontrol?
So the question is whether or not just biocontrol alone could be sufficient for glasswing. We don't know, honestly. And the issue is because we don't know, although glasswing isn't all that efficient a vector, we don't have thresholds. It's really hard to develop this kind of like population thresholds or treatment thresholds or whatever for insect vectors. So it's hard to get an answer for that. And honestly, it's been really hard to even document conclusively the impact of biocontrol. It's all basically kind of correlative or anecdotal, right? You see declines and then, oh yeah, there's parasitism, but linking the two has always been a challenge. Carmen, did you want to jump in?
Just wanted to mention that there was one of the successes of the program that I mentioned in Chile. because thank you. What happened in Chile was that the Easter Island that is far away from the continent was hit by the glass wind sharpshooter. It was found that there were route of the airplanes coming from California, Hawaii, going to Easter Island and then landing in Santiago, Chile. So the industry in Santiago was really concerned because in Chile, in the Easter Island, they were seeing this insect that they didn't know what it was. So the program was already developed in California. So it was a group of us that went to Chile, and it was this kind of a crater. It's an amazing place where the sharpshooters were, and there was no access to people or machinery or spraying or anything. So Mark Huddle had this reading of those insects that were already known that they were parasitizing the glass wind sharpshooter. They were released in many quantities, and it was very successfully controlling glass wind sharpshooter. So that way, the wine industry in Chile prevented the surge of this glass wind sharpshooter coming from Easter Island that was making all the route from California all the way to southern of the hemisphere. So that's the only testament that we have that the natural enemies can control. But remember, no insecticides at all. So that would be probably the only way that the natural enemies could be successful. If we think about the management that we have with so many other pests that we have facing, we have all the mix of different crops, it makes it difficult. But yeah, there is one way that it has been shown that the biological control was successfully controlling this pest.
Yeah, thanks, Carmen. Any other questions?
From the chat?
We have one question
Yeah.
from the chat about, can you go back and revisit organic sprays, please?
I'll do it in the next slide. How about that? For the question about the chat about organic sprays, I'll touch on the next one. So if you go to the UCIPM website related to conventional treatments, as I said, the kind of backbone is a spring application, one of these systemic. So things like imidacloprid, thimethoxam, dimetephrine, clothianidin. So the trade names, I'm bad with trade names, but it's what Admire, Platinum, Venom, Belay maybe, yeah.
Should you rotate those for preventing potential resistance?
So should you rotate?
Those are all neonicotinoids, all
Yeah.
part of the same classification, and it's not recommended to do them back to back for resistance sake.
So there's no benefit to rotating among those.
Yeah.
And unfortunately, the only systemics we have for classifying. Yeah, I believe that's still the case. Only one, there's some stuff that's potentially could come on label, but right now they're all in the index. All the same mode of action. Certainly if you are gonna do one of a late summer foliar application and avoiding clothianidin and avoiding the neonics and using a pyrethroid would help, it'd be recommended. Anyway, I kind of touched on this already, but these products have high insecticidal effects, and at least we know from imidacloprid, there's an anti-feeding effect we discussed as well. And they have this long residual effectiveness. The main point is not waiting too long if you're gonna put it on, right? Because it will take a week or two to get up, and it's usually applied as a soil drench. So doing that in the spring, sometime ideally in March, early April, if possible. And that again should protect vines for 3-4 month period at least. I struggle with this one. I don't typically recommend a later application unless it really is a kind of a massive secondary peak occurring. And the reason being is what I talked about before. You got to work around harvest oftentimes, since the post harvest intervals can be really tricky. Something to consider.
On that note, is it best to late summer after harvest? Or
So after harvest,
during harvest?
yeah. So
Because for practical purposes, it just doesn't fit into most people's. schedule.
No, it doesn't. So you have to have enough warning to do it ahead of time or really after realistically. After though, I really question it because then you're getting, it depends on the variety obviously, but
And
then you're getting well into late in the season where we don't really worry too much about any insects flying around
the
infections.
other is, you know, what's the easiest way to go over and test and determine if Pierce's is still present?
So that's maybe
Is
a
it
question,
visual inspection?
visual
If you have
inspection.
10,000 vines, it's a lot to walk through.
So the other question is basically confirming PD presence. Visual inspection is still, it is time consuming, and it's a big piece of what we do. And the other trick is that's usually you're working around harvest for that as well. Because you ideally want to do it right before harvest, because some of the symptoms are actually the clusters and what's going on in the clusters. So it's just one more thing to add to a really busy time of the year, unfortunately. And then as far as testing, I'll maybe leave it up to Philippe and others, but like there are definitely some labs you can send out samples to, especially until you get really comfortable calling symptom a symptom, which can vary a little bit among varieties and age of vines and stuff like that. And at least running some samples through a lab is not a bad idea. But I'm an entomologist, I don't know about these things. Okay, so back to the organics and then we'll wrap up. The issue with organics, there's kind of twofold. One is that we only have really two products, and really the main one of these are these pyrethrin. Pyganic is, I think, the main trade name. They have a really short effective residual period. They're essentially contacting insecticides. Even within a few hours, certainly by a day or two later, they're not really having insecticidal effects. So what that means is you typically have to do a series of these things. It's spaced out every 10 days or so. And collectively, those can have a big impact. For those of you guys who are organic, who do use pyganic treatments, they're not cheap. They have to be applied right. And so really kind of getting the timing of them and doing them honor to make sure you start things and kind of wrapping things up at the right time, it becomes really important. There's a pile of information about those kind of treatments that are out there. The other one, which I don't have as much experience with, are kaolin clays, so like Surround. You guys may be using it for other purposes. Like sunburn, I guess, sunburn control and other things.
Trees,
What's that? Olives, okay,
fly.
for fruit fly?
Yeah.
Okay, so it does the same thing, probably we're using it for an olives and fruit fly with respect to sharpshooter, based on table grape research up in the Central Valley, where they found applications of it. Basically reduce attractiveness. The insects can't sense the volatiles of the plant, and they also don't like the texture, and so they stop feeding and don't feed as much. I don't have as much experience with it, especially here in Southern California. But it's definitely another thing you can consider, in addition, perhaps some pyganic applications. Again, relatively short-lived, though, it might require multiple applications. So I'll wrap up there. I appreciate you guys' attention.
Thank you.
Yeah, question.
Distance of spread and rate. So if you have an infected site, what
How much
part of
worry?
this, right? Yeah.
Yeah. The question's basically how far the glassine probably can move PD. It's a good flower by sharpshooter standards. The natives really, you see the blue structure up in Northern California, Napa, it's really a riparian species. You get the strong edge effect. It's like the first 50 vines or something. Doesn't tend to get out in the middle of vineyards. Glassine, that's not as much an issue. And so the early days when we're getting epidemics of PD was sweeping through the entire vineyard. I mean, there's a edge effect, a gradient in from citrus, but over time the entire block goes down. Hundreds of meters is basically the short answer.
In a season.
Yeah, absolutely. Like in short order. Yeah, within a season for sure.
There's several questions online.
Yeah.
I'll answer some of them.
Sure.
The first is, was it encouraged to spray proactively regardless of insect sighting, or wait until we see evidence? Can you comment on the phasing of applications based on tracking this?
Yeah. So I think you guys can read it on the chat about insecticide applications and sort of when to do it. Prophylactic treatments are really never a good idea. Kind of going back to your question though, once you get familiar with where and when glassing pops up in your area, you can probably start to predict somewhat comfortably where, if it's there, how abundant it is, stuff like that. If you look at the UCIPM website, basically the treatment threshold is a presence of a sharpshooter, which is uncomfortable, makes me uncomfortable, but that's sort of the reality of what things are. Because again, goes back to kind of what Philippe was sort of touching on is is we don't have thresholds, treatment thresholds for these vectors, and so that's not a bad treatment threshold, the presence of it, until you get more comfortable with it. We did actually some of that work, the survey work I talked about, where we scouted for PD related to sharpshooter populations in Temecula. At those low periods when we were doing it, 2012, 2013, 2014, populations were super low and prevalence was pretty low. And we actually, some back of the envelope calculations suggested that the treatment, the cost of treatment, especially a series of organics, may not be worth it at those very, very, very low levels. But until you know what that is, what those levels are, or how many you're likely to see over the season, is pretty tough. So the quick version of is prophylactic treatments, no, but if you start to see any insects, especially early in the spring, even starting pretty soon now, then it really does argue that you're likely going to need to do something later in the season.
And from your entomology background, is there somewhere that someone can look into more of the kale and clay properties for anti-feed?
If that person contacts me, I can send out a PDF of some work. It was research done by USDA folks in the I think, Bakersfield area. So I can send some papers to them.
So please reach out.
Yeah, so my email, by the way, I think it was up front, is mattd @ucr.edu.
And then Jennifer asks if you may be able to answer this, what effect does the spraying on neonics have on human consumption for the wine? And is the organic spray any safer to use? It's tricky.
Yeah, no, I can't give a good answer, honestly. The neonics get into the do get into the fruit, get into the berries, but especially by harvest time, they tend to be incredibly low. So it's not regulated by any authority in a meaningful way. It's not viewed as having an impact. And then are organic safer? Is that the question, basically? Poorly or wrongly applied organics still not a good thing either, though. So just keep that in mind. And that's true for everything, right? Making sure it's applied right in terms of timing. Like the organics definitely are softer in general, but if you apply it at the wrong time, you're still going to hammer pollinator. So it's still, you still have to pay attention to the label there. And then that includes residues on the fruit and things like kaolin, for example, that will leave residues on the fruit. And so there's prep involved in clearing that off as well. So yeah, generally softer, especially to some of the beneficial insects. But if used improperly, it's still bad.
So if I can add on the neonicotinoids, I have a background in development, product development. And the neonicotinoids are actually based off of nicotine, found in cigarettes, cigars, etc. They're very, very, insects are very sensitive to nicotine. They're very sensitive to the synthetic neonics. And they do have a degradation curve that all companies have to apply to toxicology work, that there's a degradation curve and then an additional safety factor. And that's why you can't apply them up to a certain amount of days per pre-harvest. So the degradation curves and the toxicology work required by the federal government are very important. That's what provides the safety with those materials.
Great, thank you. I think what we've heard so far was everything mentioned, but Pierce's disease, for those of you that don't know, was first found in the late 1800s. It's also known as Anaheim's disease, where there used to be lots of grapes and there aren't anymore. And of course, we all know Ontario and Cucamonga and areas like that, that when we were kids used to be thriving regions. So it's pretty exciting to see the progress that's been made, especially in the last 20 years since 2000 on this problem. It's very devastating, clearly and certainly. There's no grapes in Anaheim. There's very few up in Ontario and Upland, and we saw what it could do in Temecula in that regard. Matt mentioned this project on monitoring. That's very commonly these days called citizen science, where we as people and individuals can help contribute to the process and help advance knowledge quickly and rapidly. I encourage always everybody to participate, even though it's a hassle, participate when you can. It just allows people like Matt to leverage their programs that much quicker. You could take something that's maybe a 5-year study and turn it into a 2 or 3 year study if you get enough participants and enough data points. So citizen science is really progressing rapidly everywhere, and it's great for adults. It's great for kids. Get them involved. So I think we're going to take a 10-minute break. Why don't we try and come back at 10:20, little bio break. Pro tip for those of you that don't know, there's also bathrooms upstairs as well as outside the doors here. All right,
And food.
what's that?
Snacks.
Oh, and snacks and refreshments. Thank you. And in order to do that, I'm going to introduce my newest friend, Dr. Philippe Rothhausen, which I butchered his last name, but I knew I
Actually,
would.
that's really good.
That's pretty good, huh?
Yeah.
Pretty good. So Philippe is in the botany department at UC Riverside. And which is right across the way from the plant pathology department where I got my degree. And so we have a lot of things in common here, and I'm really glad that you could make the trip down and talk to us all about Pierce's disease. So Philippe.
All right. Well, thank you for having me. Yeah, I have a bit of an identity crisis because I'm a pathologist, but I work in botany and plant science. You know, that's always a conflictual there. So we're gonna pivot a little bit. You heard a lot about the insect. I'm going to focus a little more on the pathogen, the causal agent, the bacterium. Although with all those vector diseases, you can't really talk about the pathogen without talking about the vector, so they go hand to hand. So there'll be some redundancy with the previous speaker. That's okay. All right, so that's the map showing the distribution of the causal agent, Xylella fastidiosa, the bacterium that causes Pierce's disease on grape, but it actually goes to many crops. You see it goes to almonds, citrus, olive, plum, coffee, and peach. Now, there's different strain of the bacterium. So the causal agent Xylella fastidiosa. On grapes, it's subspecies fastidiosa. So that strain goes to grapevine and almond. And so like the same pathogen will go to almond and called almond leaf scorch. It goes to as well citrus and olive. It is a different subspecies, Xylella subspecies, Xylella fastidiosa subspecies falca. We don't have that in the United States. This is a quarantine organism, and we don't want to have that organism in the United States. Just to give you an overview of what it does on other crops, this is olive quick decline in Italy. You probably heard that. This is a major problem now in Europe, because you can take down an orchard just in a bit of time, just like it does here on grapes. It does the same thing in olives in Italy. And then you have almond leaf scorch here in Spain, where it has become also a huge problem. So when you talk about the pathogen, it's always about the threat, because it can take down any crop, olive, almond, grapes, in just a couple of years, a few years. And this is where it's becoming a huge issue, because there's not much you can do about it. So again, this was the headlines back in the 90s. It devastated the grape penicular area, and like we heard before, because of that interface, citrus and grape interface is the main issue in penicular, so all down south in general. And you see, it's very hard to make any grapes when you have a vine that looks like this. So that's the pathogen. It lives in the vascular system of the plant. In green, that's the vascular system, that's the xylem. So that's the xylem elements. It moves water and nutrients throughout the plant. And so when the bacterium is inside the plant, it multiplies and looks like that mac and cheese aspect in the vascular system, where the water and the nutrient cannot actually move. So the plant starts to choke, it's become weaker and weaker, and that the bacterium multiplies, eventually kills the plant. So some of the symptoms that are typical to Pierce's disease, we always refer to the scorching. This is not working on the screen under the concrete. So that's the scorching on the leaf. If it's a red variety, you will have that red margin. If it's a white variety, the margin will be yellow. So it's sort of specific to red and white cultivar. So that's, I would say, this is the most typical symptom. Now, there are others, those green islands, for instance, you have uneven maturation of the periderm that happens late in the season. If you see something like that around August, September, this is also a typical sign. Reasoning, So the berries doesn't develop just because, again, you don't have water that goes through the berries, so it dries out. And of course, going with this, and match petioles. And so this is also another example. So you have the petiole that stays on the cane, and the leaf drops. And so it looks like that match samples. This is where the disease is currently in California. And so you see, it's everywhere. And like we mentioned, it's been identified in Anaheim in 1890, so a long time ago. So the disease has been here. We've been dealing with it for a long time. And it's been vectored by all those sharpshooters that Matt was talking about. Now, the issue came when the glassy wing sharpshooter was introduced in California. It's native of southern eastern US and northern eastern Mexico. This is a problem because if it moves up to Napa and Sonoma, then this is where we have a bigger problem in our hands. And so again, we'll talk here about the potential threat of the disease. Now, there is resistance of insecticide. We've talked about it, and that's an issue. I talked about it with resistance in the San Joaquin Valley, but also the climate change is going to have an impact on the vector. It's going to change the geographical range of the vector with a longer season and broader geographical range, but also with the vine recovery. When you have cold weather, when vine go into dormancy, they can actually recover from the infection. So if you have a higher temperature, they are less likely to recover from potential infections. So that could also become a problem. So when you look at the Northern California and Southern California, there's a very different pattern of disease. And we talked a little bit about this. And again, in Northern California, it's mostly edge roads that are affected because of the insect vector that are present up there. They don't have glass-living sharpshooter. We are down south because of the citrus grape interface and the ability of the insect to build up to a high population and perhaps fly further out. It's, the entire vineyard can become infected. So you see a different pattern of infection between north and south. And so that's what it would look like in Northern California, where this is the edge of the road here, where you have the first 5 rows where you have missing vines because of Pierce's disease. But down south, you will see something like this. All those vines in the middle are replaced because of it. So very different pattern when you look at it. So the state response, well, the first thing was to contain the spraying, and regulating the shipment of plants here is critical. And so here, this is the data from showing the number of plants coming out of nurseries. You see most plants coming out of nurseries are free of disease. And so this is a good thing because over 50% of nurseries are located in Pierce's disease area. So you want to make sure that when you plant a vineyard or a vine, it is PD free. That's the first step. And so this is the case. So that's a good thing. Now, survey and detection with the spore traps, right? You show that slide. This is a beautiful slide, but this is a scary slide when you see that, right? You don't want to see that. So you want to make sure that you trap the insect in your vineyard, make sure it's not present, and the rapid response is key. So wide area management program where there are sprays at a large scale being happening to keep the population of the insect low. And also, like we talked about, release of the predator to keep the insect population low, especially in residential areas, is also very important. The outreach program is also key to educate you about what to look for and how to manage it. And also the research that we discussed is very important.
The question.
Yes,
We
sir.
have an online question. If you know something about how many days below 40 degrees it needs to be to dissipate the disease, are there any studies on that?
No, so there hasn't been a lot of research done on the matter. It was done by Bruce Kirkpatrick at UC Davis a long time ago when I was a graduate student, so late 90s, early 2000. There might be a little more now that come out, but people haven't been really following up on this. So, and I'm not up to date on that literature, so I'm not gonna be able to answer that question. Matt, do you know about this?
I think you're right, like, there's still work going on now, there's not like a clear cut number yet, more is better, it's basically consensus, such as Bruce and others have shown, like, and 40 degrees is the number. Whether that number is exactly right, how many days, we don't really know yet.
Yeah, I think it would be good to have the answer to that question, but funding is always the bottleneck to everything, so. So vector management. Matt talked about this. This is how you have to manage the vector. This is to managing the disease. And so imidacloprid, neonicotinoids, or organic alternatives are good. And so I have to change that. And I heard you said May, June application. It sounds like it should be earlier than that. And so you have learned a few things today, so that's great. So probably April is a better time for neonicotinoid applications. So that's actually, and that's a study that was done in San Diego County way back then, and maybe Carmen, you were involved in this, but you see they basically spray a vineyard with neonicotinoid and left another one unsprayed, and you see what it looked like after several years. So there's a very huge difference between disease and vine removal, different vines. PD-resistant variety, that's the other option. And those PD-resistant vines are now available on the market. This is the breeding program from UC Davis. That's Andy Walker. And so I think this is a great option for planting resistant vines on the edge, especially next to riparian areas. I've talked to winemakers about it. So it's 97.5% Vitis vinifera, 2.5% Vitis arizonica. And because Vitis arizonica is resistant to piercing disease. The average consumer of grapes won't be able to tell you the difference, to be realistic. If you're like a winemaker, they can tell the difference. They'll tell you, I've talked to folks and they say, we don't want to plant it because it changes the quality of the vine. Well, because they are expert in the matter and they can tell the difference. I wouldn't be able to tell, and I like wine, but it's not there.
Could they just avoid harvesting the vines?
Yeah, or they can make a separate batch or something, but this is why I think it's a great option in the edge row, because those won't be affected, and you don't have a buildup of the pathogen inside your vineyard. So I think this is great. When it affecting your entire vineyard, maybe not such a great option. I don't know. You're
The
the one
whites taste
to
better
decide.
than the reds.
Oh, you had them?
Yeah.
Yeah,
The
okay.
reds have a little bit of a purple tinge to them.
Yeah, okay. Anyone tested them? Okay, go ahead.
I wanted to mention that Andy Walker, when he brought the samples and they were blind samples, of course, I couldn't say which one was the one that with the PD resistant or not. But I think the issue with the PD resistant varieties is that he's suggesting to use them for blending. He's not making point that you have one bottle, particularly one of them. So, and they are great for mixing them in with other. So it's not thinking that it's going to be bottled and labeled as that particular varietal. So I think that's what is being helpful also for areas that are really heavily heated by the PD and have no other option. And there are areas in Texas that this has been the survival tool for them to continue the industry alive.
Thank you.
Yeah, that's a great point. Like, you know, there's other area in the US, like southern and eastern US, that's really impacted by that disease. And so they are now they have options to basically grow grapes, which is a great thing. So that's another tool in the portfolio to manage the project. Yes.
We have questions online. Rootstock is resistant or the entire plant? Are they grafted? You know, it's top, bottom, or the whole thing?
It's the scion.
It's a scion.
Is the science. So they have transgenic rootstocks and that there's research being done on this. And the idea would be that the rootstock is transgenic resistant to PD and the scion isn't. And the resistance will be systemic throughout the plants. This has been going on since I've been a grad student at UC Davis, and I still haven't seen, you know, anything coming out of that. So maybe hopefully one day we'll see it, but there is ongoing research on this. And there's always, you know, the people are kind of reluctant to plant transgenic grapes. There's always that aspect of it. So it's more of having the technology available just in case, you know, we are dealing with an epidemic eventually. But I just don't think this will become broad stream. All right, so now I'm going to talk a little bit about my research program and other people's research program as an alternative to Pierce's disease management. So we've looked at it from a microbial perspective. Like how can we manage the pathogen, the bacterium that lives in the vascular system of the plant, with other microbes that are present in grapes? And so just like you do with probiotic and prebiotics to human, we want to apply the same concept to agriculture. So I'm going to talk a little bit about that. There you go. There are several biological control agents to the pathogen, to the bacterium. We heard about biological control agent to the vector, to the insect. There are also some that are controlling the pathogen itself. So some were studied at UC Berkeley by Steve Lindahl. This is a bacterium that originates from onion in Europe. And so you probably question like how that might be effective in PD. Well, it is. It works to some level against the pathogen and able to control the pathogen down in grapevine. There's also some studies done by looking at a benign strain of Xylella fastidiosa. So if you inoculate vine with strain of Xylella fastidiosa that are not virulent, you trigger the immune system of the plant. And so when you have a virulent strain of Xylella fastidiosa that comes, then it's already immune to the pathogen. And so you have a better response of the plant against it. And so this is also being done with John Hopkins, right? Right. So I didn't realize that you only on there, man.
I took the photo.
Oh, that's why. You're the guy. Other option currently available. So those two other options are not commercially available. This one is commercially available, and Israel is going to talk about it in a second. XylPD. This is a bacteriophage. So a bacteriophage is like a virus to bacteria, if you will, where it is very specific to bacteria. And so what the phage does, it comes and it's lysing the bacterium itself. So it attacks the bacterium and kills it. And so you have to inject it to the vascular system using that device. And I'm sure it's right here, and we're going to talk about this. I've used it myself. It's actually very friendly. It's very easy to inject it. The problem is like the limiting factor here is like if you have hundreds acres of vineyards, then you can't really do that. But for small growers, it's actually very feasible and manageable. And so I've used it and it works. So our approach was to looking at the community of microbes living inside grapes. So we were specifically interested in vines that look like this in a vineyard where you have Pierce's disease, where some of them die and some of them survive. And so our hypothesis was like, well, the other microbes that live inside the vine are antagonistic to Pierce's disease. So we wanted to know which one were those microbes. And so that was our hypothesis that we tested. So when we looked at the microbiome of vines, we looked at several tissues and from the cane, the sap, the root, and all the organisms associated with the roots. So those are, all the different tissues we looked at, and then we sampled vineyard from Northern California to Southern California and look at what were the microbial communities associated with grapes. And so when you look at a vine with very mild infection to severe infection, then as shown here, you have a biological response to it, a microbial response to it, where this is a measure of microbial richness. So as when the vine is mildly infected, you see an increase in microbial richness inside the vine, indicating that microbes are there is some sort of response from the microbial standpoint against the infection that is happening inside the vine. So we were interested to see like which one of those microbes were antagonistic to the pathogen. So we basically sequenced tissues, and we found that, well, inside the vine, this is not a very microbial diverse environment. It's a very poor environment compared to, let's say, the root system. Like this is data from the cane tissue, and we found that actually there's only a handful of organisms that are present, and especially this one. So now I had to actually remove the name of those organisms because there is intellectual property on those, and so I cannot disclose which one of those organisms. But bottom line is that those two organisms that live in the vine were negatively correlated with the pathogen. So in other words, when they were highly abundant in vine, then you couldn't find the pathogen. Okay, so they were antagonistic this way. And so what we did next was to re-isolate those organisms and culture them and reintroduce in the vine system and see if they could mitigate Pierce's disease in some way. So we grew those organisms. I'm walking you through the assay, how we do it in my lab. We grow the organism, inject it in the vine using like a needle inoculation technique. And then 12 weeks later, we measure symptoms and we measure how much bacterium, how much pathogenic bacterium we have inside the vine. Okay. And so this is our assay. So we rate vines based on no symptoms to a lot of symptoms, as shown here, and everything in between. So and so we are able to rate individual vines and rate individual treatments. And what we see is that when we reintroduce those beneficial organisms into the vine, we have a reduction of symptoms and reduction of the pathogen inside the vine. So this was very promising and very interesting. And we got a lot of interest from the private sector to look at this in the field. So this was picked up by the PD glassy wing sharpshooter PDGWIS board, and we had a third party look at all those biocontrol agents. So I talk about the biocontrol from UC Berkeley, from Steve Lindahl, the bacteriophage, and our biocontrol, to have them to do a comparative study done by a third party, UC Davis, and see which one were performing better. So this is Akibe Escalon, he's an extension specialist, just like Matt and I at UC Davis. He's conducting those trials. We all give him our strain and say, all right, do the experiment and tell us which one works, basically. And so you might have seen this. This came out this year in the Wine Business magazine. This was also recently in the PDGWIS bulletin talking about it. So if you look at it, it's present online. You can read about it. But I'm going to walk through some of the results here. Just to conclude, so we had, here you see all the different treatments to the left. And then you have And in orange, this is when we did the infection with the pathogen. So it was done in May, which is, we try to mimic what's happening in normal field conditions. And then we had several treatments, a negative treatment that didn't receive the pathogen. And so this is the rating of the Pierce's disease. Here, you see there was no PD rating. This is here the PD rating on the left. No PD rating, but then when you introduce the pathogen to the system, you have a high percent of disease, which is expected, and this is what we want. Now, the different treatment that we had were the Xalfi-PD as shown on the label, and the label indicates that you have 3 injections. Three injections after in the summer, and you see there was a reduction of the Pierce's disease symptom using the commercial product, telling you that this is actually working. So if you use it tomorrow, you would also, or this year, you would also have an effect. Now, we looked at the different treatments. So we had all the biocontrol here. And when you look at the biocontrol effects, they were working to some capacity compared to the control. But some of them work better when we did a synergy injection between the biocontrol and the bacteriophage, because they're working at a different capacity. Okay, so some of those biocontrol are bacterium, and the Xalfi, the bacteriophage. So we wanted to see if there was a synergy between the two of them. And this is what you're looking at here, is the synergy between those organisms, biocontrol and Xalfi treatments. So bottom line is that the biocontrol works. Okay? You have a commercial product that is available to grow, that is currently working, and we are working with the private sector to actually try to improve the formulation and perhaps release a new product on the market that would work better than the one we currently have. So I think this is, I look at it as a successful story, because when you're a researcher and you try to develop a biocontrol agent, a lot of it never see the daylight on the market, right? And here, there's a potential for something that comes out of the research and actually makes it to the market and will be available for you eventually. But probably you will talk about that later. So, okay, well, I'm happy to answer some of the questions. I just want to acknowledge the person that did the research here, my graduate student, Ella Dayet. You can see Israel is going to be the next speaker here. Israel was my graduate student. He actually didn't work on Pierce's disease when he was in my lab. He worked on other crop systems, but now he will move on to bigger and better things. So I will let you speak next. But yes, I'm happy to take some questions that you may have.
It sounded like your research is going more towards a maintenance, an annual maintenance, as opposed to a resolution
For maintenance for
PD.
well,
It's still there. You stop treating, it's gonna come back.
you all the idea is like just like with insect, I think you will always have to spray or to inject the vines to be able to manage the disease. There's no silver bullet, like so like if you have vines that are infected with the pathogen, you will have to continually treat the vines with either your insecticide or with your biocontrol or both for it to work.
I guess what I'm trying to get at is the benefit, cost benefit between just ripping out and replanting as opposed to continual maintenance and some of the problem that we know is treated.
Well, this is a cost benefit analysis. I think like when you plant a vine, you would want your vineyard to manage your vineyard as long as possible. So you have a return of investment. If you pull the vine out within 5 or 10 years, I think it's probably might be difficult to have a return on investment. So by keeping PD down either by insect treatment or, using those options like a biocontrol agent, you will extend the longevity of your vineyard and have the return on investment. So it is a business decision on your end, What we are doing as a researcher is try to improve the tools in your toolbox to manage the disease. And then it is your decision you decide like which one you're going with. But with PD, the problem is like in 2 years, you can take your vineyards down very quickly. And in such case, it is at cost you often. So did I answer your question?
Yes, sir.
Okay.
Yeah. What's your best guess on where the original, the beneficial microbes, like, are they getting into the vines in the nursery, or are they kind of patchy distributed in the vineyard? Do you guys have any idea kind of where they might originally be coming from?
From the roots, actually. So everything is moved from the roots up in the plants. And so, it's true for grapes, but for any plant, whatever you do to the soil and the root system will translate to the entire plant. And so plants recruit microbes. They are not passive, they are active. And so they're actually recruiting microbes in their environment to cope with stress, disease being one stress. And so I think that those microbes that we see inside the cane of the plant they come from the cordon originally, but they are vehiculated through the sap and, from the root through the sap up in the upper area of the plant. So there is when we talk about terroir and identity of the vineyard, what you have in your soil matters because it will translate to the canopy of a vine. However, there's also historically what happened in the nursery also matters with respect to microbes. I'm only talking about microbes. So like microbes that come from the nursery are also lasting inside the vines because we're talking about a woody system. So it's a perennial system, and like those microbes that are inherited from the nursery, some of them stay. Some of them are pathogens, unfortunately, but there is also some beneficial microbes that are there and that are here to stay. So there is that dual aspect of it in terms of microbes in the inheritance from the nursery, but also what happens in your vineyard is also matters, so yeah. Does that answer the question?
Yeah.
Yes, sir.
In your trial, you had 3 different biologicals. You had, I guess, one that you isolated, one that Steve Lindau did, and there was what was the third one?
We have 2 actually in our from our research. So 2 from our research, one from Steve Lindau, and one was the bacteriophage. So that's actually 4 of them. And the dual inoculation or the synergistic effect that we're looking at is the bacteriophage with the bacterium, just because they have a different mechanism of action. And so we were interested in looking at that dual effect.
And does the bacteria go down as well as up when you inject, or do you know how it distributes?
So we don't have a lot of information. So it doesn't move very far in the plant. It moves up in the plant, but not very far. The data that we have, we're able to trace it inside the cane of the plant, and it moves, maybe up to 5 nodes. So if we wanted a systemic effect, we need to do, several injections. But what's important is that how they act on the plant, because some of them actually stimulate the plant defense. And it doesn't matter where you put them, because as long if this is like how they work, then you just have to put them in the plant and they will trigger the plant, and systemic is the response.
Well, earlier we were looking at a slide where a late season infection that plants can recover.
Yeah.
And if you treat a vine and pathogen count goes down, you never really eradicate it. Does it come back following season, or how long does it stay disease free?
So yes, you never really get rid of it. So it's a lot of it would be, for some of those microbes, it would be a preventative treatment, right? But the phage is actually a curative treatment because you need for the pathogen to be here in order for the phage to work. The phage cannot thrive if there's no pathogen because it needs those bacterial cells to multiply and kill them, okay? So this is curative in that sense. So if you have a vine that is infected and inject the bacteriophage, it will reduce the pathogen population down and feed on it. Whereas the others, they don't have the same, they work in a different manner. And then you would need to introduce them into the system in the plant before they are they are infected with the pathogen. So it has to do with mechanism of action on the plant. Yeah. Yes, sir.
From last year's symptoms and looking at the plant, I think I have some Pierce disease plants. How soon should they be removed? And is there any way to save the plant?
So we have monitored infected vines over 3 years in Temecula Valley. And we were thinking that all those vines that were infected would die after 3 years. We've seen some die, but not all of them did. And some actually recovered from the infection. So I think we don't have an understanding of that. So as a pathologist, I would say, take your vines out now, because this is a source of inoculum for your entire plant. So if you have a vine that's infected, and you have an insect that comes and feed on it and goes to the vine over, you will basically transmit the disease over, right? So you don't want to have this. But from a disease standpoint, some vines, from our experience, can recover from the disease, you know, so it is, I think it really depends like how far gone they are. I think there's a threshold after which like the vine will not recover and will never recover. But if it's a mild infection, I would say try the bacteriophage, try to treat the vine with the bacteriophage and see if you can get recovery with this. Because you don't, in my opinion, you don't need to take the vines out if the infection is mild. If it's severe, it might be too late. But if it's mild, I think there is a hope now to cure those vines from that infection.
If it is mild, what do you suggest I do?
Use what they're gonna talk about. And I think this is, right now, this is the best option. Once you've done, you know, if you manage your insect and everything, and you have, the last hope for you is to try to cure the vine from the current infection. And now you have tools available to do that. So I would say try it and then see how it works.
Okay, I've taken a few cuttings and planted them. Will they be infected if I cut them from a vine that has the Pierce?
Well, you do, yes. You don't want to propagate from infected vines because you will transmit the disease. So that's, you want to do it from clean plants. Yes. Yeah, and not only Pierce disease, like other diseases as well. It is better to, you know, viral disease and bacterial disease and whatnot. You want to start from clean stock, and nurseries are usually the best bet for that. Not every vine is certified free of all those diseases out there. It's mostly viral, Xylella fastidiosa, but there are other diseases that are not on that spectrum. But I think it is a better and safer way to go to start from clean vines from nursery rather than try to propagate from your own vines in the tree. Comment? Go ahead.
Yeah. Really,
Go ahead.
I need this. Thank you. Could you please mention if you included in your trial the vines that have been already received systemic insecticide? So to be sure if there is any effect of the chemistry of that has been already in the plant using all the, what we potentially definitely with the treatments for the insect, if there is any effect on this bacteriophage.
I don't think it has. So it's the assay is done at UC Davis, and I don't know if they have treated for the insects. Well, they wouldn't be the glassing sharpshooter there, but for the other sharpshooters. So I will ask, but this is a good point, like, because this is only the first year of the experiment. And there's like 2 years behind this to make sure this is consistent. And we get good results like that for year 2 and 3. So one thing that we can add is actually make sure we have insecticides, right? That's your suggestion, right?
Yeah.
Yeah, okay, all right, that's a good point. All right, thank you for bringing that up. Yes.
Is this being done in different cultivars? Because it's been shown that different cultivars have different thresholds for foliar symptoms.
Yeah, no, it's only done, this is on, at UC Davis, this is on Cabernet Franc. And so this is, yeah, and they have that because it is a good variety for viral diseases. And so that's the only one we could work with. From our experience, some varieties are more tolerant to PD than others. So like Merlot is a more tolerant variety to PD than Cabernet Sauvignon, for instance. If you move to table grapes like Thompson Seedless is very, very susceptible to the disease. And so this is a not good variety to plant usually. So there is, and the mechanism of tolerance to PD is not very well understood. There are several components to it. We think we've done some work on this. We think it has to do with the size of the xylem vessels. That's one parameter. So Merlot has very close, small xylem vessels, and it tends to be more tolerant because it can respond faster to the infection. Whereas vines that have large xylem vessels, like Thompson Seedless, it's a very large xylem vessel, so you can move water quickly, is more susceptible to it. But there's also like the bacterium. When you look at a cane of a vine, xylem vessels are different segments to one another, and they're connected by pit membranes. And the water can move through that, but the bacterium has to move through this as well. And so like the chemistry of the pit membrane seem also to matter in terms of the tolerance, because they have different chemistry in some varieties. And so some bacterium, it takes more time to break down the pit membrane and move to the next one. So there is different aspect to it, but we don't understand. There's a lot of varieties out there, and we've only studied a handful in terms of tolerance. We don't always understand, to give you advice at which variety to plant. We don't always know which one is tolerant, which one is susceptible. They have only studied the main ones. So I think that would be valuable to know, to have that understanding of which one are tolerant, which one are susceptible, from my standpoint. Other questions? Do we have questions online, or we're done?
The question online that came up was, can the vines that had PD, if they're in recovery phase, be a source of Xylella? And I responded, unfortunately, yes, because you did mention that a few minutes ago. And then the next topic on Xylophy PD will help, I think, bring to light a little bit more detail on what treatment and what to expect from it.
So the next topic, sir, will be helpful to answer the question about which disease is going to be. Okay. Desire? That's over there.
I went through that one, so. Oh, I forgot. No. Something? What that?
Well,
All right, so thanks, Philippe, that was great. Now to continue on the discussion, we're going to move into the Xylophy PD system and brought to us by Infotech and some work. I think
Israel will probably explain where this came from and how we got it here. But it's pretty exciting news, I think in the PD control front, and we're really glad that Israel and Josie could join us. So Israel.
Thank you for the introduction. So Israel Luna and my colleague, Dr. Josie Hughey. So we all come from A&P Infotech, a company which is based in Palo Alto, California. So today we're gonna explain pretty much how our product works, right? And just gonna go over it kind of fast in the beginning of the slide, since our colleagues already mentioned some, history about that. So briefly, the Pierce disease overview will be quick. The history of bacteriophage or bacteriophage, depending on how you want to pronounce it, and then the efficacy of it. So how, it started in Texas A&M in 2015, they did the research trial, and the efficacy tested by UC Davis, and then provide some results from commercial growers from 2019 to 2021, and then some of the feedback from the growers too. So we know that PD can be the limiting factor for wine production, right? Typically, the infection occurs in vines that are 2 to 4 years old, and it started in California in the 1800s, right? And it has increased in California in the recent years, mainly in Napa and Sonoma. So UCANR estimates that there's a 56.1 million loss every year, and the CDFA has put a lot of effort to control Pierce disease, but so far, right, the focus has been in the sharpshooters, which are the common vector for PD. So this is a most updated map from CDFA. So from 2023, the locations in that are colored is where PD has been reported, and it's mainly in the sites that where vine production happened. So now moving on to the biology of Pierce disease. Okay, so we call this our villain, right? We have our Xylella fastidiosa bacteria cell, which has these long tails, they're pilus, right? And then we have our air transmitted by the vectors, the glassy wing sharpshooters, and we already this, the blue, green, and the different types of sharpshooters, right? So Xylella fastidiosa colonizes the gut of the sharpshooter, and then the sharpshooter will feed on the sap of the vine and then transmit the disease. So here, Philippe already mentioned, right, that once Xylella fastidiosa enters the plant, it will form these biofilms, and the plant basically has this vascular system that conducts water, right, known as the xylem. And one way for the plant to respond against the further expansion of the disease is by releasing these cells that are called chyloses. So the too many chyloses can start, clogging up the vessel and causing water disruption, and that leads to the mimic of water stressing plants. So as you can see in the leaf. And then this is just a table of how there are other diseases out there that can be also confused with PD. One of the main ones is red blotch or Eutypa dieback, and there are many others. Okay, so Philippe already mentioned, right, the leaf scorching. So the top row is for white grapes, and the bottom row is just for red grape. So it's pretty much right, form these concentric zones, which are a series of rings of leaf dying, or the margin pretty much dying, right? And then the what we call also the petioles fall off, and then we have all these matchstick petioles. And in this picture, the berries, they are dehydrated, they shrivel, right? And then we have that that pattern, strange pattern of irregular lignification. So pretty much parts of the stem kind of hardens. And then, yeah. So there are some molecular tools in order to do sample testing. So some of the companies are Agri Analysis, ALNL Crop Solution. So once those symptoms appear, which happen to be in May to late season, right? Just take some petioles and from the cane and send them for testing. So they use this method known as qPCR, which is a method just to detect the presence or absence of PD. Briefly, there are different types of control for PD. So we have the classic one, the most common one, the physical control, right? It's just inspecting your vine for the presence of PD, right? Pruning, trying to prevent PD, frogging or replanting as well, and placing your vines away from sources that can be for vector or the pathogen in this case. Other controls are the vector, so they already mentioned here the trapping and monitoring of the sharpshooters. Some insecticides that do not work that well, and parasitoids, Matt already mentioned about it. So, and then the transgenic and PD resistant vines, right? Although they are resistant, they can still get PD over time. And then we have our bacteriophage, which is what I'll be talking about in the next slide. So we have our hero here. So to our bacteriophage, right? It's a virus.
They're really big.
Yeah.
Wow.
So I have something here. It's our right. So this is our bacteriophage, and then it has a capsid, which has the nucleic acid, and well, it has also the long fiber and spikes at the bottom, and pretty much how it works is that for the figure on the right side, the phage will attach to Xylella fastidiosa. It will inject its DNA. It will replicate inside of it, and then it will burst the cell, and more bacteriophage will be created and look for more Xylella fastidiosa. So basically, bacteriophage was discovered over 100 years ago, and they have always been proposed as an alternative, right, to control pathogens, and they are very specific, right, harmless to people. And this approach for PD will be used as a biocontrol agent, right? So here, just a closer look. The Xylella fastidiosa type has this tails, the pilus, and then the phage will attach to it and penetrate the cell. The next slide, this is a closer look. Once it enters the bacteria, the virus will replicate and create more of them. So, and we'll seek and try to destroy more of the Xylella fastidiosa cells. So now transitioning to how this was discovered started from Texas A&M in 2007, but here they just collected some samples from wild grapes, and then they cultured it. They isolated those Xylella fastidiosa strains. They sequenced that and then created a bacteriophage library, pretty much, which is what we have in our product, a cocktail of bacteriophages. So this is our product. It's a 100 milliliter bottle or vial here, and it's organic, so it's certified by OMRI. And here in Southern California, it's distributed by Kalina, and any part of Napa will be by the Wilbur Ellis. And we have our guest here, Jose Huerta, if you wanna stand up and introduce yourself really quick. You need a microphone.
Yeah, this is, we're, Adam Glenn, we both work for, Wilbur Ellis, we're distributors. So if you have any questions on, how to get this product, we'll be able to talk to you.
And then is there any members of Helena in here this morning? Okay, so we're missing one of our distributors here today, but Wilbur Ellis, if you guys are interested in the product, the two representatives from Wilbur Ellis are here today to speak with. Thank you for joining us.
So, sulfide PD can be used as a preventative treatment or as a therapeutic. If it's in vines of less than 3 years old, it can protect your vines from getting PD. If your vine already has PD, you can treat it and still keep it alive. So we already said that we can use the product in areas where your vineyard is surrounded by riparian areas or with high sharpshooter counts. So here is how we have our product. Let me show a demo. So it consists of our gun. Well, actually, let me do my vest. So we have this vest, right, to carry our sulfide product. And then it consists of the CO2 canister. So it's attached to a hose, right? And then, so the gun has a needle, right? And then inside of the barrel compartment, that's where we have our product. So we have a syringe pretty much, and then this use tiny hose to extract our product. Insert that into here, then with the knob adjust pretty much the dose that you want to use. It's basically between 60 to 80 microliters, so it's a tiny amount. And do the injections at the base of the vine, and depending on how severe PD is in your vines, you can do 2 to 3 applications per season. So just want to show you, it's pretty simple to deploy the product inside of the vine. And this is just water, so see. Good. So if we don't have the CO2 canister, there are other portable ones. Just the one that Josie has. And I believe with the portable ones, you can do up to 1,000 injections. With this one, I think it's 5,000 injections per vine. Yeah.
What does it cost for the smaller one roughly?
The CO2? the smaller, that's just the CO2 for the propellant. The product only comes in one size at this point in time, and the distributors right behind you can give you the price.
I think it's asking the unit
Oh,
itself.
The unit itself, so this is actually this is leased through a company called Pulse that makes the injector gun. And so it depends. So one of the things that's really been this is an area where we've had some really novel approaches, where we have some smaller growers that only have 200-300 vines. And there's a gentleman that wishes he could be here this week. He actually got a whole neighborhood group together. And what they do is they lease one injector per year, and they share the equipment and share the product. Also ensuring that it's cleaned between every use to make sure there's nothing transmitted from one location to the other. But if you have a group of neighbors that are struggling with Pierce's disease, it's a really novel approach to work cooperatively. Because the bottle itself can stretch 300 mature vines or about 600 for young vines. And so if you don't have that many, find a partner or some neighbors and try to work together as a group for the injector. And then, of course, the price is available through the distributors that are right behind you. The Xyloject lease each year is around $768. And then if a group of growers come together and purchase 10 or more bottles, then a lease injection is included for free. Any questions? Yes, Carmen, question.
If you tested with the likelihood of carrying the bacteria with the injector?
So far, there have been no reports of any movement of the bacteria with the injector itself, because the outward movement of the product from the needles outward is dispelling it. There's also one grower, for extra caution, out of Napa has suggested that they use a lighter if they know they have a diseased vine, and they'll use a lighter to sterilize the needle before injecting into the next vine. So I think it's good to steward as best possible, but the transmission with the equipment itself has not been reported severe. Thank you. So, Israel, would you like to continue your discussion? We're all giving the wrong thing. Yeah. Let's cover that, and then we'll let you continue on the others. Maybe see, let's see. Maybe this is the equipment. She's switching the camera over. So for the online people, they're the eyes.
Yeah, they saw it.
So just to continue, right, they did this testing to see how far the product can travel along the vessel or the xylem in a vine. So they use a food dye, and it can travel to 10 centimeters from the site of injection within minutes. So how do you actually apply the product? So if your vine is less than 3 years old, to protect it, you do 2 injections at the base, both in opposite sides. If your vine is a mature vine, then you do 4 injections, the same 2 injections at the base and 1 on each corner. And it's recommended to do anything between 60 microliters to 80 microliters. The application starts once the plants break dormancy and start flowering. So you do the first application, and then the second can be done after 4 to 6 weeks. And depending on the PD pressure in your vine, if it's too severe, you can do up to 3 applications per year. So briefly, just want to talk about the Texas A&M greenhouse trials in 2015. So this is a treatment where they use to treat PD prior to Salmonella fastidiosa. So in purple, we have our treatment, and in orange, we have our inoculation. After week 3, they conducted the study for 12 weeks. It was 10 here. And then they also did the treatment to compare inoculation and the treatment, right? And the variety was Cabernet Sauvignon. And after 12 weeks post inoculation, they look at the visual assessment, or they tested the visual assessment. So the picture on the right is only phage, right? So it's clean. Then Xylella fastidiosa inoculation, then you can see some of those symptoms, right, that are chlorosis, stunting, and matchstick. And the treatment of vines treated with sulfide PD and also inoculated, well, they don't look that bad either. So here is a graph of results. No cases of PD detected using sulfide PD, which is shown here in blue and green lines. And there were 66 fewer cases of PD using the product as a therapeutic. So in red was just the Xylella fastidiosa inoculation only. So now from UC, yeah, sure, question?
Going back to that, yeah, so just to clarify, we see 66 fewer cases, so of those vines that were confirmed infected by Xylella before, 2/3 of them no longer tested positive by qPCR, is that?
Right.
Online question real quick is the shelf life of the product. So I'll answer that one if it's okay. The shelf life of the product can last a couple of years or over a year if it's refrigerated. It's not recommended to store it at room temperature. Sorry, the shelf life of the product. It can be stored for over a year in refrigeration. It should be used fully within 36 hours once it's opened. So try to use up a complete container at one time because you don't want to risk contamination. Open it, use it, and then, but you can store it or pre-order it for the season up to a year in advance, as long as it's refrigerated.
So now moving on to the UC Davis field trial. Philippe Rosalsen already mentioned this. The project leader here was Kip Escalent from UC Davis. From UC Berkeley, we had Stephen Lingo, and the funding was obtained from the PD board. So Philippe Rosalsen also contributed to the project. So you already mentioned this table or graph or figure here, but I will only focus on the first 3 rows, the healthy control, the disease positive, and the sulfide PD. The rest are trials that might work down the road. So they started their inoculation in May, right? So it's I believe here 16 weeks. So we have our healthy control, we have our disease positive in the second row, and then we have our sulfide PD label, which is inoculation, and then followed by 3 applications of sulfide PD. So from that study, it's after visual assessment, polar symptoms. Here we have in red our disease positive, and treating the vines with sulfide PD, it almost reduced by half the polar symptoms. So that was the main conclusion from that study. And then from transitioning to commercial vineyard trials from 2019 to 2021, it was a 3-year study. And it was here in Sonoma County, and 3 different sites were selected, actually 4, right? And then there were 3 different treatments: untreated, treated with sulfide PD for 2 years, and treated with sulfide PD for 3 years. And just to point out, these sites were located next to a riparian area, so, and they had a high PD pressure there too. So from 2018, they have no visual symptoms of PD, and then in 2019, they started this study, so they have 3 different treatments, right? The untreated, treated with sulfide PD for 2 years, and then for 3 years. So they did their applications, right? 3 different applications, every year from 2019, 2020, and 2021. And the final result here was evaluated by qPCR and by visual assessments. So the graph on the right, are vines that were PD positive and were tested by qPCR to detect the presence or absence of PD. So we have our control, right? And then if you treat your vines for 2 years, that reduction of PD can be lower by 24%. And if treated by 3 years, it's by 45%. Then the graph on the right, it's more visual assessment. So the visual symptoms are reduced by 27% if treated for 2 years, and by 54% if treated by 3 years. Yeah. So conclusion from here is that there are no new infections in young vines if treated for 3 years.
How many treatments in each of those 2 or 3 years during the season?
3.
3?
Yeah.
Yes.
Now, if you look at, from the fruit yield standpoint, right? So treating your vines for 2 years that have PD, they can bump it up to 10%. If treated by 3 years, it's up to 17%. So our product works, right? It shows yield in symptomatic vines that were recovered. There's new fruiting wood that was established from vines, cut down to the base, and there's a reduction in replants, right? And well, yeah, moving on. This could be used as a preventative protection for new plants. So now let's look at a couple scenarios, assuming that you have PD, right? So here in yellow dots, if you have a vineyard, right, that has PD and it's by a dry creek, right? So you would want to treat your areas here in purple box, right, to create a buffer zone and prevent further expansion of PD into your vineyard. Now, if you have, scattered vines that have PD, you treat them, and also the vines surrounding the infected vine. Now, in cases where PD is everywhere, pretty much have to treat the whole block with sulfide PD. So in summary, our product, right, prevents or preserves the health and productivity of vines. This is a cocktail of bacteriophages, right, that will attack Xylella fastidiosa. And we got excellent support from the Integrated Pest Management. It's OMRI certified, and we since it's our organic product, there is doesn't require a lot of PPE or equipment. So you just grab the gun and inject your vines. And from all those field trials, right, there's 99.7 disease free. And then you can also increase your yield by 17%. And here is just a QR code in case someone is more interested about the educational programs about PD and on. So you can go to the AGCEU website. And yeah, with that, I can try to answer as many questions as I can. You guys have some.
Yes, any possible way of using this liquid in a backpack? Is spraying the vine?
That is the most common question. Israel, thank you for allowing me to join you up here. I joined the here, so I wouldn't trip over things, and I did anyway. Thank you, Kate, for getting this back online.
Go ahead.
All right, so the question was, is it possible to use a foliar version of this product? It is the most common question that we are asked, and certainly one that our development staff is working very hard. They're working, and it's a difficult process because these bacteriophage are fairly large and they have to find an effective way to get into the plant. And so, yes, we are having some good results so far with a foliar version, but that's a few years into development. We hope that.
2 years.
Understand. Well, and that's why we went ahead and moved forward with the direct injected phage so that we can solve problems in cure Pierce's disease now. So. Question.
So what about using it through the fertigation system?
Yeah, that's
Is that the
another.
second most popular
Yeah,
question?
second most popular question is, can we put it through the drip? And that is also, but the complexity of soil texture, soil adsorption properties, and would also take a long time for development work. So what the benefit of having the direct injection now is that it's putting the product directly into the xylem, which is where the bacteria colonize. So you're basically getting perfect coverage with where the disease is infecting the plant. And so we understand that this is sometimes a big barrier for people, but and we've got quite a few effective applications happening in commercial vineyards now where people understand, like, they're really trying to just recuperate their yields. Israel? Want to take any more? Next question.
Is there any projection in the future that maybe we wouldn't have to treat 3 times a year, every year? Because if, down here, the pressure is everywhere. We're not like Sonoma. And so if you have 50, 60 acres, that's like more than one person full time who would be gunning every vine.
I want to tackle this when you want me to. Okay. Yeah, that's okay. So Israel joined us a month ago, you guys. I think he did an excellent job.
Yeah.
For coming up to speed quickly. If we can give him a quick round of applause. So that's also another great question that we get is, do I have to use it 3 times a year at the max dosage for multiple years? What we're finding in earlier studies is that you can actually see a reduction in the amount of Xylella presence after several years of use. There are some initial studies that we have that detect the bacteriophage in the insects and the vectors. So they can actually pick up the bacteriophage from vines that you injected into. And we are also finding the bacteriophage outside of the vines and things like weeds. So there's, while the work has not been published, there is an indicator that you can reduce the amount of Xylella in the surrounding areas that would be where the vectors are, picking up the Xylella and bringing it to the vines. Another question.
So with the gun, because you mentioned the leasing of it for a year of $700, is there an option coming up soon to buy one?
Yes, you can buy one, and the pricing, Israel would be the right person to contact for pricing because you can lease it for 1 year, 2 years, or purchase the equipment. It's under $3,000, I believe, to purchase the gun, and of course, I think there's probably a service cost if you send it in to have it cleaned up once in a while. But you would work with Israel directly on any equipment needs, and he would work with Pulse and help get you in contact with them.
What's the recommended buffer? So if you have a hot spot of something in your vineyard and you do like 8 around it, or if you're a bigger area, you should be.
Yeah, so question was recommended buffer, and let me talk about a buffer zone treatment first. Where one of the users where we have blue-green sharpshooter in the Sonoma area, they saw Pierce's disease up to 8 rows in. They're treating 12 rows in now from the riparian area.
And they're going all along that riparian area, so they're giving themselves an extra 50%. Now with the spot treatments where you see a diseased vine, one approach is to do one vine all the way around. And then there's a grower in Arizona that's actually treating the 2 adjacent vines and everything around, so he would be more than 8 vines around the treated in that case. So it's really about understanding your pressure and from a trapping standpoint or from just your disease history and making the right plans for each vineyard on a case-by-case basis.
Josie,
Yes, sir.
question is, it's probably more a question for Matt. Because if you're dealing with the blue-green sharpshooter in Sonoma, does the same buffer treatment and
No.
treatment around it infected play out with GLIS being infected?
No, you're different. So it's part of it's the spatial pattern issues that
You
you're.
wanna,
As Josie was saying, like, there's a strong edge effect, blue green, and so. There's a couple of things,
let's start a panel.
Why don't we just move into like a little panel discussion? Any question for any of the speakers? Philippe, if you want to come up? Sure. Carmen, I don't know if you have anything to contribute. Anything that you want, but better to have all three, 4 of you up here.
So there's a couple of things with PD and Zalella. It has a relatively long-ish latent period, so the vine may actually be infected, you may not see it. And from what we can tell, sharpshooters can actually pick up the bacteria from the vine. It doesn't look diseased, at least a few months, if not more before. It's kind of tricky to know for sure. So doing some kind of prophylactic, what looks like a prophylactic thing, at least on adjacent vines to an infected vine makes sense to me, sure. And in blue-green areas, the strong riparian edge effects, I could also see that makes sense to me because we see these strong gradients away from there. And so treating certainly the vines that are showing symptoms, maybe some of them around them and maybe an extra row or 2 in, that I get. Temecula down here, it's just gonna be different. And so treating in a whole vineyard or even many rows of a vineyard where you're not seeing any symptoms maybe doesn't make as much sense for us because of the way glassing moves. You'll still be patchiness, and so definitely treating injecting some of the vines around the ones that are symptomatic makes sense. Doing the whole block or many rows of the block, I'd like to see a little more kind of evidence data first.
I think what I would like to add to your comments, Matt, is that what we saw in Temecula, when we had those huge numbers of insects, was when the insects were colonizing already in the vineyard and the risk of the nymphs walking to the next vineyard. So that's the one to vine transmission. What would be a factor that would be different if we have the other sharpshooters that are more prevalent, like in the Napa area.
Yes, we have a question in the back left, Mr. Jose Huerta.
How long does the bacteria phase last in the system?
Repeat the question.
He asked, how long does the bacteriophage last in the vine? So. Yeah, so it's 6 to 8 weeks, so that's why you have to do another application after 4 to 6 weeks.
Thank you. Other questions online?
Yeah.
Okay.
For those of us online, is there a ballpark idea of cost of one vial?
Would our distributor in the room like to speak up on the cost of product per vial?
Yeah, I think it's about $1,000 a vial, which comes out to like $333 a vine, treatments.
Okay, for mature vine.
Mature vine.
Okay, so remember, there's 4 injections for a mature vine. There are only 2 injections for a young vine, 3 years or younger. You want to repeat that for the online crowd?
Yeah, so the question was the cost of one vial, so it's around $1,000, right?
Yeah.
And you will have to contact Wilbur Ellis distributor, and so that's for younger vines, you only need 2 applications. For mature vines, you need 4 applications. And depending on the severity of your PD, you can either inject 60 or 80 microliters and apply it either 2 twice a year or 3 times.
So when you're talking about 3 applications per year and 2 injections per application, can you go to the maximum 80 microliters per injection and go through a vineyard faster by doing it twice rather than 4 times?
Great idea. So right now, we have a commercial user in Sonoma that addresses your question. And your question, as I heard it, for the crowd over here, is, could we just increase the dosage and do 2 injections instead of 4? If you think about the vascular system on the vines, the 4 injections on a mature vine, 2 at the base and 1 in each one, is really about distributing the product where it needs to be to help clear the disease out of it. So on young, shorter vines that don't have a lot of wood up top, then the 2 injections is sufficient for full coverage inside for the vascular system. As a vine ages and becomes 3 or 4, 4 years or older, you're going to want the injections to be closer to the wood that can be infected by the green tissues. So it's about distribution of products, not necessarily just the volume alone.
Yeah, just want to add something to this. We have to keep in mind that when the vine is infected, the xylem vessels are included. So if you inject anything in it, it's not going to be, it's not going to go systemic. So it is important that you go to several places to make sure that the vines picks it up and goes into the transpiration stream. So it goes systemic in the vine. If you do 1 injection in a vine that's already included because of infection because of other diseases, it's not going to go nowhere. So you have to do, by doing multiple infections, you make sure that actually the bacteriophage is distributed in the vine. So it is very important.
Your question up front?
Yeah, how many injection sites do you recommend on a mature head train?
Head train is interesting. We have some diagrams that we can send to you for mature head train vines. But essentially you would want to rotate which of the arms from a head train that you would inject within the season. If you have noted symptoms on one arm as opposed to another, then start with those. Because the vascular system is going to track along with each of those arms. So certainly protect those that you're aware have symptoms first, and then rotate throughout the season with the upper injections. Yes, sir. Question at the back.
About $1,000 a bottle, I seem okay, but with a 36-hour one's open lifespan, is there any plans to make smaller bottles? If we need, let's say I need 1.1 bottles, I'm gonna lose $900 every time I have to use it.
Yeah, so the question is, okay, what do I do if I need just a little bit more than 1 bottle? And we have discussed internally at the company about making a smaller container. So we'll provide that feedback that that's important to you, and that's certainly a possibility down the road. For right now, where other growers are where they need 1.1 bottle, this is where the neighborhood collaboration started. And it's a grower in Escondido that has 250 vines in his backyard, but 1 bottle, with his vines, they're mature vines, and 1 bottle can treat 600. And so he has a neighborhood consortium that he's gotten, friends and neighbors together. They purchase and inject together also, so that they're planning efficiently with their
Not an
product
option
usage.
for where I'm located,
Yeah.
but
Okay, yeah.
we're also mostly probably winemakers. We're pretty good at keeping oxygen out of stuff.
Yeah.
What is the issue with once open?
So once you open the container, because it's a biological system, this is the phage are living beings. And because once it's open, there's a risk of, if you remove some of the product to put it into the injector, you can introduce bacteria, you can introduce, whatever's in the atmosphere around. And the purpose for not having a long shelf life once it's open is to prevent any contamination from becoming a problem upon injection. So that's why it's encouraged to use up a complete container at once. And certainly for your particular vineyard, we can help work with you on just a lot of the.
Putting a different cap on it, so I can just take it out with a syringe would be.
That's a possibility too. So I think maybe this is a great conversation if anybody has case by case basis questions like this, because the product's fairly new to the market and we have growers of various sizes. Reach out and we'd be glad to talk with you about your specific scenario. Cool. Yeah. Right, Israel?
When you talk about injecting at the base, so as far, you gotta get on your knees and do it at the bottom, or you can?
Yeah, so when we-
It basically be interpreted a lot of.
When we talk about injecting at the base, it's going to be above the grafting point. And yes, it does, this is not a back-friendly administration of a product at this point in
You gotta get a
time.
little rolling.
Right, I very much encourage proper ergonomics, be safe with your back. But yeah, we've got
Just above the graph.
just above the ground. We have growers that are.
The ground or the graph?
The graft, right above the graft, sorry, I said ground. We have some growers that have cut their vines down just above and they pulled new wood from above the grafting zone. And they're treating probably 4-6 inches off the ground.
So that makes a case for the Uber vines.
Yeah, I wish I had a little slit that just moved you forward from one vine to the next.
I think they have something like that.
I have one, I have one.
Another question, yes.
Safety, what happens if you accidentally inject yourself?
If you accidentally inject yourself, I'm sure that the label prohibits that. But so these bacteriophage, as both Philippe and Dr. Israel Luna mentioned, these the bacteriophage are specific for Xylella fastidiosa. So it's hard to know exactly what all the effects would be if you were to ingest it or if you accidentally injected yourself. But it certainly would attack any Xylella fastidiosa that you would have in your system. So, but they are, these bacteriophage are found in nature, and they're naturally occurring, and that's why we are able to get the OMRI certification because they exist as very naturally occurring things, and they don't have impacts on other bioflora or microflora. But I think it would, it probably would.
If one of my field hands does that, do I send him to the hospital or do I tell him to see?
Well, it depends on how bad he's bleeding from sticking himself with the needle, but I don't think there's a major risk unless there's contamination from the vineyard somehow that got into his wound. I would say you would probably want to treat it on a case by case basis. The product itself is fairly safe. Yeah. We have an online question, yes.
Are any commercial nurseries looking to use this as a preventative in new vines?
Sorry, I keep stealing this one. But the question was, are there any nurseries looking to use this commercially as a preventative? And we've had some inquiries, but we certainly need to follow up. If they wanted to send their contact information, absolutely, we think it'd be a good partnership and it makes a lot of sense.
Are there any commercial nurseries that are actually using it and promoting that they're treating?
Mm-hmm. Are there any commercial nurseries that are using it currently and promoting that they're treating? Not that I'm aware of. We have some discussions with a couple of nurseries, but I'm not in contact with them at this very moment. I'm also fairly new with the company. I came on board last fall. Our tech rep last year decided she wanted to go back into the wine industry, and so any questions that we have, certainly I'll dig as deeply as you need to get the answers for you. Yeah, was there a question?
Philippe wants to jump in.
Well, I just wanted to, it's, when the scale of production of vines at nursery, it's not a system you can put into a nursery pipeline just because you can't inject every single vine in nursery. It makes sense to inject the nursery vines, however, the mother vines, because in that case it makes sense, but like every single cutting is not feasible. Now, when your company comes up with foliar application, then in that case it makes sense. But other than that, we just see it being feasible.
Yeah. One final addition, the closest that we have to that kind of use in the field is there's a vineyard management service company in Sonoma that had potted vines that were transpiring actively before they planted, and they injected them prior to planting. And so that was one of the novel approaches that came out of 2022. Now, to your question, yes.
I'm a QAL with probably over a dozen clients. And our routine has been injecting imidacloprid sometime around April. Which is really great because we really like the systemic process as opposed to foliar or walking. I have a lot of clients that are concerned about the effects on bees with imidacloprid. Anybody care to comment about that? Is there an alternative to imidacloprid?
Nothing that works in the same way. And it would just be these foliars which have their own non-target impacts too. There are reasons to be concerned about mimics and pollinators as well as birds and other things. Grapes are actually one of the lower risk uses though, because they're not pollinated, right? They're not insect pollinated. And so the bigger issues are sort of timing especially can get a little bit trickier around fruit trees. In grapes, we don't tend to worry about it as much. And again, like any kind of spray, there's potential for drift and other impacts. So if imidacloprid, I think someone was asking about it, what happens or if imidacloprid is going to disappear. If it does disappear, it's going to be replaced by other stuff used in the short term that arguably have at least as immediate impact, especially if they're misused on pollinators and beneficial insects. So I'm not going to tell people not to be concerned about it, and I still fully support folks using organic approaches as well. We're not seeing insecticide resistance in Southern California yet with imidacloprid, and compared to some other stuff that's available, as long as it's used properly, I think most of the risk is mitigated to pollinators.
Thanks, man.
Well, yeah, I was just thinking about that, with the halo cells that start to close up the xylem once the plant gets infected. So does that restrict the flow of the bacterial phage to distribute to the vine?
That's what Philippe was exactly the point, yeah.
I missed that point.
And this is the irreversible. So once it happened, then the xylem vessel is not biologically active anymore. Now, vines keep making wood and new xylem vessel every year. So as long as it keep making some, it's fine. But yeah, once the vessel is shut down, that's it. Now, if you have embolism like drought, you have cavitation in the xylem, this is reversible. So if you put water back in the system, it's become biologically active. Thiosis, that's it.
You belong in the body.
Well, I have to do some of it.
I think I would like to make a comment. I was doing some research regarding irrigation, and this is widely known. And there is those dendrometers that are attached to the cane, the trunks. And it was very interesting to see the cycles, how the wood expands and contracts. This is for the company as just being curious. If would be interesting to see if they follow that cycle and probably find that there might be some times during the day. And this actually we were seeing that the trunk expanded at night. That might be more efficient and it's cooler and probably the distribution, it would be better. It would be interesting to see a comparison, understanding the cycles and in regard to the irrigation cycles as well. So that probably will give us some interesting information around that.
Thank you, Carmen. So there are recommendations on the label for timing of application with respect to when the xylem are active. So if your vascular system has not yet begun waking up for the day and it's cool, and it's not sunny yet in the mornings, your vascular system is not moving water through the plant and distributing it very efficiently at that point. So it's recommended to wait till later in the day when the xylem movement and the water movement through the plant are actually occurring for the injections. And so there's also guidelines on the label that tell you that right after a rain, when the ground is saturated, the xylem isn't pulling a lot of water up at that moment and right after an irrigation. So I think knowing that the xylem needs to be fully active, it's when the vine is thirsty and it's pulling water from the soil is the best timing for application. Thank you, but I like that I did tracking it.
Other question. I was emailing back and forth the other day with Hailing Jin, Dr. Hailing Jin. For those that don't know, Hailing Jin is the professor at UC Riverside that isolated a microbiological active peptide from finger lime that controls Wong Bing disease. And our preliminary results indicate that it does the same as Pierce's disease. So I don't know if you've heard that or you're aware or no, but I think it's a very interesting new addition to the story.
Yeah, that's right. And I know I've heard she's requesting funding to look at it in the PD system.
Yeah,
So it seems to be applicable to grapes as well.
yeah, she said the preliminary test that she's done in greenhouse and outdoors has indicated that it's got the same activity against Xylella that it does against Wong Bing.
Yeah, this is correct.
So from the system point of view, it sounds like it would be really interesting to get that.
Although they are probably
Maybe
years
next time we
behind.
do this, we can have her come too and talk about it.
Well, because you would be treating the citrus that's right next to the grapes. So it would really-
Yeah, I don't know much about that peptide and how the application methodologies come into play, but you know, there could be synergies between all three, the biological,
Uh-huh.
the phage, and then this peptide that she's discovered.
So I would like to return to the question that I asked before about the risk of, or concern of carrying the bacteria with the injections. And when I was working with Andy Walker and his way to develop the resistant varieties, he brought a pure culture of Xylella fastidiosa with thousands of individuals, and we're injecting. And I just want to let you know that the transmission is a very, very complex process. And to this time, I don't really know if this is understood. This probably is already published. The bacteria seems to need to have something additional when it's already being harvested in the guts of the insect. We were not successful as the rate that we expected, having the injections directly with the pure culture of bacteria in the field. We needed to have the insect to be efficient to be able to transmit it. With this, I have no data, I cannot tell you what percentage is, but I think it reduces, in my mind, the concern that with the injection, I will be carrying it. Because it seems that the bacteria itself, even if it's a plant that is infected with the pathogen, being in the syringe, the level, that would be the titer that probably that I need to start the colony in the next one. So it was interesting to mention that the one that was concerned with even adding an extra lighting on the tip. But to this point, I think we need to remember, this is a very, very complex process. And Mother Nature has equipped the insect to be able to carry, that's the bacteria what needs to be additional powers in the gut for the insect to be able to inject it in the plant. So probably we will be, hopefully, with no data with this, but you already mentioned, you already have seen it. We will be protecting really, that will be achieving the goal that we are looking for. Thank you.
Any other questions?
Has this stage been done with sharpshooters?
So the question online was, has the phage been found in sharpshooters? And yes, there are preliminary unpublished studies that show that the sharpshooters can ingest the phage. And we've also found it in other vines that weren't treated with phage. So there's suggested data that's been collected that they can not only pick it up, but they can move the phage, which over time can help reduce the amount of Xylella present around the area after several years of use of Xylella PD. However, that work has not been published or peer reviewed at this time.
Anything else, anybody? Just before we wrap up, in case you didn't catch it, Unit of Continuing Education, through that QR code, if you need it, zap that. And this was a good session. I appreciate everybody for showing up and giving such great information. If there are other topics, for those of you that are in the room or online that you're interested in the future, shoot us a note or let us know if you want to shout it out now. We'll make a list and we'll see if we can put something together in the future. It's good, great crowd. I don't know how many people are here. 30 to 35. And I understand we have over 40 online, so great participation. We appreciate all of you showing up and learning what's new. So thank you to our presenters and thank you all for showing up.
Well, we'd only need like.
Oh, yeah.
Okay.